geographical research is wherever sample is involved and is testing on the sample, statistical tools are used. So here I'm going to explain some simple method for statistical data analysis, and with the help of Excel, how can we analyze data that I am going to tell here. So here, data analysis in biological research can transform its result into useful research outcomes. So generally in biological research, medical research, data analysis is important because outcome of that data analysis will decide whether your product or your study is useful or not. So on the basis of outcomes, researchers can make informed decisions, generate well-formatted, testable hypotheses, design follow-up experiments, and provide compelling biological and mechanistic evidence for results. So here, let us see, suppose this is population. And in that population, there are a number of subjects. Now, population means population of subjects. Now, subjects can be human being, so population of human being, or population of woman only, or population of gents only, or population of men below 18 years or above 18 years, or it can be population of crops, it can be population of insects, like this. So it is a population, and we have to study some characteristic of this population. Then that population may have large count, maybe lakhs of subjects or millions of subjects. So you cannot study every subject of this population. So we take a sample. We take a sample from the population. Means here, from population sample is taken, and sample we say that it is representative of the population, means it possesses nearly all characteristics of population. Then we study this sample. Suppose we are doing experiment on this sample, then we obtain results, and on the basis of outcome of results, we can say that that experiment is applicable to this population. Now, when we say population, we have to decide certain characteristics of population. Suppose we want to study effect of some drug on high blood pressure of human being. Then we decide population, that is, it is in between, suppose 40 to 60 years. This is our population. And from this population, we derive a sample of suppose 100 subjects, 200 subjects, like this. And we study effect of that drug on that 100 or 200 subjects. We study its effect, and we generalize it, that this drug is whether applicable to the population or not. So that can be done, and for that purpose we require some statistical tools to see the outcome of our experiment, whether it is applicable to that population or not. Now here, then suppose we take an experiment. We have sample of 25. Suppose here we take simple experiment. Suppose we have 25 plants and we study effect of some pest on these plants, and we measure height of plants. Effect of that pest on the height of plants, and we get result. Then we have variables, means after how many days we are going to study, that we have to decide. So here variable, it is important one, that is height that we study. Then we got this, and if we see 1 to 25 samples are subjects are there in the sample, and we have measured height of each. You will see variation, means all of them will not have the same height. Then we find out its mean, and to find mean, x bar is mean, x1 plus x2 plus x3 on x25, because we have taken here 25 subjects divided by number of subjects. So we find mean by this formula. Then we find standard deviation. So deviation from the mean. So this is the formula for standard deviation. Standard deviation. So we find mean, we find standard deviation, but from this you may not get a lot of inference or a lot of information. So we find one quantity that is called probability. So this is a formula for probability. So here for this 25 subjects, we have, this is mean, x bar is equal to 24.588, like this. Sigma, it is equal to 7, is standard deviation 7.1386, and this is standard error, because sometimes you require standard error also. Now standard error is simply sigma upon root of n. That means number of subjects. Now we find another quantity, and that is probability. That is probability. P is equal to, and that is formula. P is equal to 1 upon sigma root of pi e raised to exponential e raised to minus in bracket x minus x bar square upon 2 sigma square. So we have already obtained sigma. We have already obtained x. So for each x, we find probability. Now, so if we find probability here, this probability P is equal to this. Now if we see here, P, it is always less than 1. It is always less than 1. If we find total P, that total P will be equal to 1. Probability of occurrence of that event or occurrence of that activity. So probability of occurrence of 23.5 is this, out of 1. Out of 1, like this. Now 23.1 can be repeated. 23.1 can be repeated. So if we suppose we have 1000 subjects, and if we are measuring height, out of that many may have same height. So depending on that, we will get value of probability. And generally, all samples all samples satisfy this probability distribution for that particular variable X. If we study any population and if we decide some variable of that population and if we study that variable and if we find probability, it satisfies this distribution. That's why it is actually Gaussian distribution. Gaussian distribution, but it is also called normal distribution. It is also called normal distribution. So why is it called normal distribution? Because normally any population, if you study, it will satisfy this distribution. And if you see here, this is the general normal distribution. So here, variable X, value of variable X, here value of probability, and here this is normal distribution. Any population will show graph probability versus value of variable, and here this is about your mean value X bar, and somewhere here we get standard deviation from mean value, and we know that standard deviation it is obtained using this formula. Obtained using this formula. Now see here, so this is average value of X, that is X bar, and see if we measure area under this curve, this one will be equal to 1, unity. Total probability is always equal to 1. So this one is correspond to 100% total area. Total area means this area. Total probability. Now, if we about this mean position, we have standard deviation, and suppose if we take same distance on both sides, same distance on both sides means this distance will be equal to this distance. So here, so if we take this range, you will have probability beyond this range also, though it is less, beyond this range also. So what happens? What is value of probability outside this range? Total probability. So this total probability, suppose it is 5%, then 95% will be inside this. If you take here this range, then total probability outside this range will be less. If you take here, so you will have certain value that separation is equal to sigma. You will have certain value outside that sigma, but most value is inside that sigma. So this is generally normal distribution, and any population for corresponding variable will satisfy this distribution. Now, what is experiment generally done in biological sciences, medical sciences, psychology, education, educational research, psychological research, even in geographical research? We take a sample, population, and we have to study effect of something, maybe effect of corona vaccine on the population, whether that whatever corona vaccine is made is useful to the population or not. So some pilot studies done first, then general studies done, but general study is not done on whole population. It is done on the samples. So samples are chosen, and generally samples are chosen randomly. So there are a number of sampling methods. So random sampling method means from population. First, we decide characteristic of population, like corona vaccine. It is for today's corona vaccine. It is for all human beings above 18. Above 18. So as population, it is all human beings that are above 18. So that characteristic is decided, and then sample is chosen. So here, this is the sample, first sample. And then second sample is taken. Sample 1 is taken, sample 2 is taken. Now, number of subjects in 2 samples may be same or may not be same, but 2 samples are taken. Suppose on first sample means we administer drug or vaccine to the sample, to the subjects of the sample. So experiment is done on this sample, and this sample is said to be controlled. Controlled means here no experiment is performed on this sample. So it is controlled. But these two are compared. These two are compared. Sample 1 and sample 2 are compared. And from whatever results of a variable, is here, suppose X variable is studied, effect of drug on blood pressure, suppose it is studied. So blood pressure, that is variable. So readings are taken for sample 1, readings are taken for sample 2. And we find here, so we can see here X1 bar, here X2 bar, here N2, here N1, here standard deviation sigma 1, standard deviation sigma 2. So mean of first sample and mean of second sample. Suppose here N1 is 25 and N2 is also suppose 25, and we get X1 bar, suppose mean blood pressure, suppose 104.5, and here X2 bar, it is 99, we can take here 102.3. Now, so we got average values of blood pressures. Now, if we see experiment, effect of drug on blood pressure, for sample 1 we got average 104.5, and for second one we got 102.3, and suppose we get standard deviation, sigma 1 is equal to 12.28 and sigma 2 is equal to suppose 6.5. So depending upon number of variables you are studying in that population, you will have number of hypotheses, alternate hypotheses. Now in inferential statistics, we try to study null hypothesis. We try to study null hypothesis. If null hypothesis is true, means accepted, then means if this is true or accepted, and this is accepted on the basis of your results, statistical results, then these are rejected. Means H1 or alternative hypothesis or thesis rejected. But if this is accepted, this one is accepted. Sorry, rejected. Then this is accepted. Rejected means no effect of experiment. It is rejected. Means there is effect. There is effect. Means H1 is accepted or H2 is accepted or H3 is accepted. So we try to study null hypothesis, and on the basis of outcome of inferential statistics, we say whether null hypothesis is accepted or rejected. Rejected on what level? Accepted on what level? That is most important in inferential statistics. So here, for that purpose, we make statistics. So inferential statistics means with inferential statistics, you are trying to reach conclusions that extend beyond the immediate data alone. For instance, we use inferential statistics to try to infer from the sample data what the population might think or what the, whatever for that population might work. Means whether that drug is effective for whole population or not. That we will draw from study of hypothesis using the samples. Now, most of the major inferential statistics come from a general family of statistical models known as the general linear model. This includes the t-test, analysis of variance, analysis of covariance, regression analysis, and many of the multivariate methods like factor analysis, multi-dimensional scaling, cluster analysis, discrimination function analysis, and so on. So these are some inferential statistics techniques. So here we have studied two samples. So before doing experiment, we make hypothesis. Hypothesis as alternate second. Now you can have a lot of samples. So you can have drugs. Blood pressure, you can study separately. Means sample 1, first drug is administered for sample 2, second drug is administered for sample 3, it is controlled. So you are doing 2 experiments simultaneously. Then you can study sample 1 with sample 2, sample 2 with sample 3, sample 1 with sample 3, and we see effect. Sample is controlled. The experimental group 1, experimental group 2, and here control group. You can compare these two statistically. You can compare these two. You can compare these two also by the same methods. So here, this is for drug 1 and for drug 2. By when we compare E1 with C and E2, if our null hypothesis is rejected, then our alternate hypothesis is accepted. Means that drug is effective. That drug is effective. Now we also study E2. So if null hypothesis is rejected, alternate hypothesis is accepted, we say that the drug is also effective. Then we compare E1 and E2. Means out of drug, first drug and out of second drug, you can say which drug one is effective, more effective. So in this way, experiments can be done. Now we will see here. So these are some differential statistical techniques. Here selection, test. Pair test. We are going to study here pair test. In the pair test, test for the difference between two variables from the same population. And here we can take pre-test and post-test. Pre-test and post-test score. So generally in educational research, pre-test and post-test scores are used. So what is the meaning of pre-test and post-test? Means before doing experiment, we study the sample, means certain characteristic of sample. Means we have two samples, sample 1, sample 2. They are selected randomly from a population. Now, before experiment, we study characteristic of that sample, means we study certain table. Then we do experiment, means here, we have sample 1, sample 2, certain variable. It is studied for this sample as well as this sample, and their variables are their mean values are compared using some test, statistical test. So suppose x1 is mean, x1 bar is mean value, x2 bar is mean value of this. This is before experiment. Before experiment, these two mean values are compared statistically. So suppose conceptual understanding of certain subject. So suppose we are studying conceptual understanding of quantum mechanics. So generally we study before experiment, understanding of that conceptual understanding of that sample. We compare them, and when an experiment is done here. Now we have to select two samples in such a way that before experiment there is no significant difference. Then we go for study. If there is significant difference between two samples, or significant difference between means of two samples before your experiment, then that samples cannot be used for study. So suppose we are studying conceptual understanding of quantum mechanics. We have two samples, then whatever conceptual understanding, and that can be done using some test. So whatever results of that test for two samples must not be statistically, means significantly different statistically. Then and then we perform study. Then we make one group and control group, and then we another group is experiment group. Then we perform experiment on this. You may have another technique for control group, or you may not perform experiment on control group. And then we take here test again. That is called post-test. So here we have pre-test, and then we have post-test. Then again we find here x1 1 bar, x2 2 bar. We compare names. If we get statistical difference, means significant difference in means, then we say that our experiment is effective or useful. So this is called pre-test post-test study. And for that purpose, generally t-test is used. Other tests are also used, covariance analysis of covariance. So these are some independent t-test. Independent t-test means test for difference between the same wave from different populations. Means here, suppose we are studying blood pressure, but groups are group and female, like this. So in this way, we use different techniques. So we'll study here t-test. T-test is analysis, and this is simple test. So see here, we have two samples. Sample 1 and this is sample 2. One of them is suppose a control and another is experimental. And for sample 1 we take mean value. We will take x1 bar and here we take mean value x2 bar. Now there is some difference. You may get some difference. But whether that difference is statistically significant or not, that can be studied using t-value. So t-value is equal to x1 bar minus x2 bar upon sigma 1 square variance or sigma 1 is standard deviation upon n1. n1 means number of subjects in first sample plus sigma 2 square upon n2. Means here we get sigma 1, here we get this one sigma. And here in first sample we have n1 and n1. So you will get t value depending on these values. Now on the basis of this t value, you will see whether difference between means is statistically significant or not. Here, n1 can be 1, n2 can be 1. Like this also. Means sample can be small, sample size can be small, can be large. Depending on that, you will get t value. So if and that value is called critical value. T critical. If your t value is greater than that, difference is statistically significant. If t value, whatever calculated t value, suppose n1 is suppose 25 and n2 is 25. 25 is another example. n1 is suppose 100, n2 is suppose 100. This is another value. So we have three sets: first set, second set, third set. So here for n1 n2 with values 25 and 25, you will have certain t critical value. Here also certain t critical value, that is also called table value. Table value. When that is standard value, that will decide whether mean is statistically different or significant, there is significant difference or not. So t, whatever calculated t may be less than t critical, may be equal to t critical, or may be greater than t critical. So that is table value. And that table value is given. Table value is given for sample set, means number of samples. Now, On what level you have to say this statistical difference? Means we study 5%, 1%, 10%, like this. That is called alpha level. Alpha level of significance. Means if we say 5%, we take 5% alpha level. What is the meaning of this? Means there is significant difference between two means of samples. Is there is 95% chance that the difference is due to experiment and 5% chance it is due to some errors. So there is 95% chance. 5% level means 95% you are sure that the difference between means it is due to experiment and 5% may not be due to experiment. So this is so you have to keep in mind this formula. You have to keep in mind this formula. Now, actually, when you are calculating p-value, p-value means probability value. P-value means probability value. Whatever value here, this is probability value. P-value. So here, by using mathematical software, statistical softwares, you can easily find values without knowing formulas. Without knowing formulas. But if you are doing research, you must know significance of P. So what is P? How is it useful to find difference? What are the characteristics of P? Whether P is sufficient to find the significant difference between two samples? That we must know before being experiment. Sometimes it happens that P alone cannot tell you whether means are statistically significant or not. Only on the basis of P value you cannot determine. Then you have to go for other tests also, like F test and so on, analysis of covariance, analysis of variance, and so on. Now here we will see what is meaning of alpha level. Alpha level means 5% or 10% or 1%, meaning this with the help of graphs. Now see here, if we see, this is x1 bar and this is x2 bar, and this is curve for sample to probability curve, and this is curve for sample 1, and variable is x. Now see here, this point. This point has same p-value, means both will have same p-value. Same p-value. Sample 1 and sample 2 will have the same p-value, and that is p-value of this calculation. We find t, we have standard deviation of standard deviation of then p-value we have calculated, then p-value we have calculated. P-value is this intersection value. Now this p-value may be greater than alpha, p-value may be less than alpha, means less than, suppose you have decided 10%. Then this p may be greater than alpha or p may be less than alpha. If p is greater than alpha, then difference is not statistically significant. But if p is less than alpha, means you have decided 5% level, 5%, but it is more effective than 5%. Means you have 95% chance that whatever difference we got is due to experiment. but we got p value less than alpha. Means suppose we got p value. Suppose alpha value is 5%, is 0.05, and suppose we got p value is 0.01. Means 99%. Whatever difference we got in between x1 and x2 means x1 bar and x2 bar, it is due to experiment. So P will show you actual value. This is pre-decided one. Pre-decided one. This is pre-decided one. So here, now see. So just I have shown you, this is p value. Suppose we take here this one. This difference, this range. Outside the range we have 5% probability, and inside this range we have 95% probability. Now, if you see here this p value will be less than this one. It is in this range. So your p value will be greater than alpha. This was your intersection between two distributions. It is inside 95%. Means difference is not statistically significant. Difference in means is not statistically significant. Now here shift here. Now here, suppose this is p value, and here also 5% here and 95% in this range. So now here you will get p value equal to alpha value. Alpha value. P value is equal to alpha value. When we have this case, then p will be equal to t critical, table value. But suppose that distribution goes like this. Your alpha level it is here, and your p value is here. P value is here. So let us here p value be here 1%. Means beyond this value, here we have probability 1% outside this. And here it is 95%. So this is your alpha level you have decided, and your p value is less than alpha. So here p is less than alpha. Then here p value, whatever calculated t value from means x1 bar and x2 bar, sigma 1, sigma 2, n1 and n2. Whatever calculated t value, it is greater than t critical. So p level or p value decides the level of significance. So alpha is pre-decided level. Pre-decided level. So in this way, we use statistical techniques. And then using p value means here we make a table. Means table like this. So number of samples n1, then here p value, number of samples means we take here control group, experimental group, number of subjects, your mean value, and your standard deviation. Here n2, x2, sigma. So we make this table. Then here suppose you can have 25, here you can have 25 or maybe more or less. Then you can have your 20.5, the mean value, suppose 8.9, here 21.1, is 12.2, like this. Now sigma 2 and sigma 1 that depend on variation of values in certain range in that channel. So sigma for some sigma can be large, for some sample sigma can be less. Now here we first know what is T critical for this one. For this one. And here label is also important because T critical for 5% level or 1% level, they are different. So we must know first what is T critical for this environment. So for that purpose, degrees of freedom is important. Degrees of freedom. Degrees of freedom. Now, what is the meaning of degrees of freedom? Degrees of freedom is there are number of independent variables. Independent variables. So suppose we have N1 is equal to 25. And suppose we know mean of these 25 subsets. Then what is degrees of freedom here? DF or DOF. Degrees of freedom. Now we mean is what? X1 plus X2 plus X3 plus so on X25 divided by 25. 25 means over here N1. N1 means you go up to here N1. 25 means you go up to here N1. You go up to N1. So here in this way, what happens? Now we know mean that is 23.5. So if we know 24 values out of 25 values, we can find 25th value. Means it is not necessary that 25th value is known. Means you know mean of this sample and you know 24 values, then you can find 25th value. So 25th value may become dependent. So here independent variables are 24. So N1 25, but degree of freedom is for 24. Here degrees of freedom. And if we take collective one now, then here this minus 1 and this minus 1. So that is also important. That's why T critical values are different. That depend on degrees of freedom also. So in this way, and then we find actual T value using formula x1 bar minus x2 bar divided by square root of sigma 1 square upon n1 plus sigma 2 square upon n2. Now this T, T depends on x1 bar, x2 bar, sigma 1, sigma 2, n1, n2. Now this T may be less than critical value or may be greater than critical value. If it is greater than critical value, then the difference is statistically significant. So in this way, use inferential statistics. And when once your T value is greater than T critical, suppose at 5% level, then we say that null hypothesis is rejected and alternate hypothesis is accepted. Means your study is effective. So you might have seen some such papers in biological science, in education. In that paper, In that article, there is table like this. And then alpha level is given, P value is given. And then they state difference is significant at 5% level or at 0.05 alpha level. So by seeing only table, you can compare. You have to calculate first, and then you have to compare. So what happens here? It may happen that two samples. So suppose we have two samples. Suppose we have two studies. Here N1 25, X1 bar suppose 23.5, and sigma 1 suppose 12.5, and here N2 suppose 25, X2 bar it is suppose 22.3, or I can say 20.3, and sigma 2 it is suppose 10.1. So what happens here? You will get distributions like this. You will get distributions like this. But if you have same one, 25, 23.5, and suppose standard deviation, it is 2.3, and here 25, 20 point suppose 25, and standard deviation it is 1.21. Now see here, what is difference? Mean values are generally same. But see here standard deviations. So you whatever here, this standard deviation is less, your distributions will be like this. Now see here, P value. So here may not be statistically different, but here it may be statistically significant difference. So significant difference. So see, things not tell you whether difference is statistically significant or not. It is standard deviations also. So this is about the test. Now we will go to some same calculations using Excel. Okay, we'll see some Excel. So again, calculator. Just I am opening the document. So see here, we are plotting some graphs. So suppose we have variation with time. So this is independent variable. So this one, so this is independent variable, and here I have taken time, and suppose height with time. This is first parameter and this is second. Suppose we want to find the plot the graph and we have to see variation. Then simply we select this one, then we press control button and we select this one and then insert, and then we use here scatter plot. So we get graph scatter plot with this. So we have point also and graph. So this gives you variation of height of a certain plant with time. Time in hours or time in days. Now what is pattern? So to study the pattern, whether it increases linearly, exponentially, or quadratically, and so on. For that purpose, we use here regression analysis. So to do that, just click on that graph and here add trend line. Now if we add a trend line, here we have number of options. If you see here, number of options: linear, logarithmic, polynomial, moving average, so on. Suppose we use here linear. So you will see on the graph there is dotted line. There is dotted line. And on this table, format trend line, here we have here display R value on the chart and display equation on chart. So here, since we have used linear, we will get equation, linear equation. You may have studied equation like this. You may have studied equation like this. Y is equal to mx plus c, where m is slope, c is intercept. So here we got y is equal to 6.854 5x minus 0.7273. So if we know x, this time you can find it. So this is general now see here you can have number of you get graph like this. So if you get graph like this, do not use linear, you can use polynomial. Suppose we use polynomial here. Actually polynomial will not properly work for this such a type of graph, but you can use polynomial. Now is it visible? There was a network problem.
Yes.
So here we got. So you have to pin my video to see the full screen, because it is not screen sharing one; it is the direct video sharing. So this is equation. This is equation. Nice. Now see, you can have bar charts, you can have other pie charts and so on. First, we will go to how to find t value corresponding to the certain degrees of freedom. Degrees of freedom I have shown you. Now here I have taken one table. This is t value table. So these are degrees of freedom. If you see here, these are degrees of freedom. df. This is level, 0.05 level, 0.01 level. Means level, we have normal distribution, and whether you have decided to study at 0.05 level or at 0.01 level. So here when degrees of freedom is 1, for 0.05 level, T value it is 12.7062. And when you are studying at 0.01 level, T value is this one. For degrees of freedom is equal to 1. What is degrees of freedom? DOF or DF is equal to N1 plus N2 minus 2. N1 plus N2 minus 2. N1 number of subjects in first sample, N2 number of subjects in second sample minus 2. Because we N1 minus 1 and N1 minus N2 minus 1, these are degrees of freedom for individual samples. So for total collection, you have N1 plus N2 minus 2. So see here, at alpha level 0.05, your T critical value. This is T table value, or it is also called T critical. T critical. So they are different. Now, if you take 2 degrees of freedom 2, it is equal to this one. If you have degrees of freedom is equal to 15, values are this one, like this. So you can find for large values of degrees of freedom, like this. Now, how can you find this value using Excel? I have obtained using Excel, and you are actually you will get statistical tables. In that statistical tables, you will get this table also. But using Excel, you can find this. So in Excel, there are number of built-in functions or formulas. Using the built-in functions, you can find values. So here we have one function is equal to T dot INV function. Now here P dot INV function, you can use two-tailed or one-tailed. Now here for such a two distributions, we generally use two-tailed. So T dot INV dot 2T. So I will take here 2T. So T dot INV dot 2T, this is function. So equal to T dot. So I will choose it. So I will zoom now. Here, I will take here T dot INV dot 2T. Now 2T, then probability. Probability means here your level. Now you can take 0.05 degrees of freedom. Now degrees of freedom, it is N1 plus N2 minus 2. So suppose we write here N1, so select this cell, plus we write here N2, so select that cell, minus 2 and complete the bracket. So this is simple formula. So we'll get T value. Now I got some num error. Why? Because we do not have degrees of freedom. So you can have 1, 1. Still we got num because N1 is 1, N2 is 1. So 1 plus 1 minus 2. So degrees of freedom is 0. So 0 degrees of freedom, it is not in the T table. So if I take here 2, then here 12.706 I got. Suppose I use here in place of 0.05, I can use 0.01. So I got 63 point. Now you can change this here. Suppose I 12, 14. 12, 14, that is 26 minus 2, that is 24. So for 24, we get this 2.5 79694. 79694. This is table value at 0.01 level. So in this way, you if you know number of subjects in two samples, so your T critical it is decided. Now calculated T value, whether it is less than this or greater than this, significance is decided on this. Now, don't pass. So here we will go to T test now. So see here, here I have table, serial number means 1 to 25. This is value of variables for control group. This is value of variables for experimental group 1, and this is value of variables for experimental group 2. Now first we have to find mean, then we have to find standard deviation, and you can find standard error also. So using Excel it is possible to find. So formula is popular, not value. Then here also. So we got average value for each one. Then we have to find standard deviation. So there is also formula STDEV. This formula. You can in new Excel there are number of new formulas. DV dot ASTDP. So I got this standard deviation. These are values. You can find standard error using sigma upon root of n1 square, sigma upon root of n2 square, and so on. Now, our value, suppose we are comparing these two groups, C and experimental, one group. So make a table. So this is first group first, group second. So n is 25, n is 25. We got average value, we got average value, we got mean value. Now, formula for T we know. Formula for T is what? T value. So T, that is x1 minus x2 mean values divided by square root of sigma 1 square upon n1 plus sigma 2 square upon n2. This is T value, calculated T value, not table value. So here we will use this formula. So suppose we will find T value here. So we'll find T value here. So T equal to, I will find T value here, SQ or deviation. This is standard deviation and we have to take its square. Divided by N1 plus sigma 2 square again divided by N2. And I got T value 3.92853. Now 25 plus 25, that is 50 minus 2, 48. So for 48, what is for 48? We have this one 2.01063 for
Hello, I'm now request Dr. Sonone sir start second session.
Good morning, one and all. Welcome to the second session of fourth day of faculty development program. It is a great privilege to introduce our today's eminent speaker, Dr. Urmila Kulkarni Kale ma'am. She is an information scientist at the Bioinformatics Center, Savitribai Phule, Pune University, Pune. Basically, she has a distinction of having contributed to and being part of the growth of the fields of the various fields like the bioinformatics, immunoinformatics, and computational virology in India. She and her group is basically working, and she had made significant contributions to correlate genomic variations with phenotypic features, and has developed several databases, algorithms, servers, which are the first in the respective categories. She has several active collaborations with virologists in India and abroad. And the output of that, she had published more than 35 papers in the peer-reviewed international journals, as well as she has jointly holds an international patent to her credit, in addition to highly successful interactions with industries as well. She is passionate about building capabilities, and she has been faculty for several national and international training programs in bioinformatics. She is a recognized PhD guide and having more than 25 years of teaching experience in the field of bioinformatics. Dr. Urmila is an elected member of the reputed Maharashtra Academy of Sciences, and has led several extramurally funded national and international projects as well. Recently, she has been inducted as a member of European Virus Bioinformatics Center, which has 178 members from 100 institutes and 30 countries. She is also a member of the editorial board of the Journal of Infection, Genetics and Evolution, and handles manuscripts in the area viruses, evolution and bioinformatics. So today it is our a very great privilege that we have received such a nice eminent personality. May I request Dr. Urmila ma'am to start our session. Over to you, ma'am. Thank you.
Thank you. Thank you very much for the generous introduction, and indeed it is a pleasure to talk to all.
You are not audible, ma'am.
Is it? Just a minute.
Yes, you are audible.
Am I audible now?
Yes, ma'am, you are perfectly audible.
Yes, ma'am. Yes, yes.
Okay.
We are able to hear. Yes, ma'am.
All right. So indeed, it's a great pleasure to talk to you all, today in this faculty development program, because you are going to be the ambassadors to take the bioinformatics forward to undergraduate students. And therefore, I thought that this is a good opportunity to interact and to introduce you with the field of bioinformatics. So I'm not sure how many of you have had exposure to bioinformatics before, and it's a multidisciplinary subject. So I'm going to, based on my discussion with Dr. Varsha, I'm going to not assume any prior knowledge, and would like to take you through this journey of bioinformatics, right from its basics and fundamentals. Of course, it may not be possible for anyone to sort of cover the length, breadth, and depth of the discipline of bioinformatics. But nevertheless, if I can enthuse you, if I can if I'm able to generate your interest in the field of bioinformatics, I think the purpose of this particular FDP would be solved. So I'm going to take you through this journey with a focus on tools and techniques, since the theme of the workshop is tools and techniques of bioinformatics. Let me just make the full screen, and all right. So, many of you come from life science background, and therefore I thought this would be the most appropriate starting point for us to begin discussion on bioinformatics. So first and foremost, biology, life science branches in biology and bioinformatics, they go hand in hand. So usually, we refer to different disciplines in life sciences as wet lab sciences, because you perform a lot of experiments. In bioinformatics, we also perform a lot of experiments, but all these experiments are performed on the computer, and therefore most often than not, the bioinformatics labs are referred to as dry labs. So, that's the basic distinction. But then, in bioinformatics, you can predict several attributes, like, if there is a carbohydrate binding site in a protein, or if a protein is antigenic or not. If it is antigenic, what are the regions that are likely to be recognized by antibodies, so on and so forth. So variety of inquiries can be made, and for all those inquiries, predictions can be made. So in vivo, in vitro, in silico. So these are three research methodologies that are currently available to us. If you have to, plan the wet lab experiments, sometimes it is very time consuming. It requires a lot of resources in terms of chemicals, consumables, etc. So probably it's a good idea to narrow down your search space using bioinformatics, and then to perform select set of experiments in the wet lab for the purpose of validation. So bioinformatics will narrow down your search space from, let's say, 100 different experiments to 10 experiments, and then these 10 experiments can be performed in the lab to validate the predictions made by bioinformatics. So on one hand, bioinformatics helps you or assists you in narrowing the search space, but there is this entire big domain of bioinformatics that deals with data analysis and interpretation. So today in the field of biology, we are generating huge amount of data. Be it genomics, be it proteomics, various different fields. But what do you do with that data? How do you harness the power of that data in order to make sense out of data? You will understand the methods and the algorithms pertaining to data analysis in the field of bioinformatics. So life sciences, has helped data generation, and it has strengthened the existing areas. Bioinformatics has expanded the domain of life science research and added very many areas for research. So life science, typically you will have omics-led projects, whereas with bioinformatics, you can make very, very fine data-led inquiries, and you can generate hypotheses, and you can design theory-led projects that can be validated. So bioinformatics, depending on the scope and the need of your project and the objectives of the project, fits into two different capacities. One is it can help you develop a project based on the data analytics, and then or on the basis of the predictions, it can aid and help in reducing the size of the experiments, and third, it can bring in a lot of predictions, and we learn from the wet lab validations of prediction. So let us say that B cell epitope prediction algorithm that I happen to develop way back. So that algorithm knowledge base was limited. There were only a handful set of antigen antibody complexes, but today there are many antigen antibody complex co-crystal structures available. So I can refine the rules of the prediction on the basis of large amount of data that is available. So the point that I want to convey and drive across is that there is no competition between the life sciences and bioinformatics. They are in fact very complementary, and they go hand in hand. And depending on your domain of research, you can explore the field of bioinformatics, inquiry based or query based. So you need not have to learn the entire gamut of bioinformatics in order to work in your area, but there are certain fundamentals which I feel you should be aware of, and that is what I'm going to cover here. So bioinformatics is a science that equips professionals with the most desirable skill set of 21st century. And what is it? It is data analytics. It is to use data for the purpose of discovery. And that is what we are going to learn here. And I'm going to explain this concept. And for that, I have borrowed this wonderful graphics from a gapping void firm, which is a culture design group. Okay, so bioinformatics helps us to analyze data, and it facilitates data-led discovery framework. So what basically is data? Data are points. Okay, I joined a few minutes before, and I heard that there was a discussion with respect to statistical analysis methods, and I'm sure statistical analysis methods would have helped you define data. So data are facts. They could be alphanumeric, they could be pictures, they could be sounds, they could be videos, they could be anything. And then that data, you layer it with the available metadata, and then data gets translated or transformed into information. And when you try to join the similar data points in a group using various statistical or data analytics techniques or data mining techniques, you come up with these beautiful linkages across data, and that is where we say that we have transformed data into information into knowledge. But it is all right, you have discovered new knowledge, but how do you gain insight? So you gain insights when two very apparently distinct or different data points get connected by a particular path, and provides you an insight with respect to functionality of these data points. So that is where you generate the insight. And wisdom is like you are using these two very different data points for the purpose of discovery, to add some new knowledge in the field, and you are able to translate when you develop that insight and wisdom, you are able to translate data-led inquiries into translational products such as drugs, vaccines, or diagnostics. And that is where we say that you have made an impact which is socially perceptible outcome of science, which is otherwise considered a very, very theoretical area of work. So you are translating data into products for the purpose of social good, because all our research is always funded by the taxpayers' money. So usually bioinformatics, the gamut or the pipeline is, or you can use the nucleic acid research. They have a dedicated issue in the month of January to describe various databases. And in the month of July, they have a server issue where one can search the available analysis methods. But NAR is not the only journals. There are large number of journals. There are in fact some journals that are dedicated to databases and algorithm development, and you can search. So you can go to the PubMed and search for the resources. You can go to the Google and search for the databases, etc., etc. So finally, the objective is, as I said, that you want to analyze data to see if there is any pattern that is hidden in the data. So at times, identifying these patterns hidden in the data is like finding a needle in the haystack. But nevertheless, it is very, very important for us as biologists, as bioinformaticians, to invest time and energy in finding out those needles and identify those patterns, associate those patterns with functions, and then search the data using these patterns and functions. But patterns can be exact. So, you have, a particular sequence which is characteristic of DNA binding domain. The patterns can be fuzzy. Okay, they are not so direct patterns. Patterns can be continuous or distributed. So you have B cell epitopes that are linear, and you also have conformational B cell epitope. So linear B cell epitopes are continuous in terms of their sequence space, but conformational epitopes are antibody binding sites. They come together due to the folding of the polypeptide chain and therefore may not be continuous. So patterns can also be regular, they could be irregular. And then we have battery of, group of methods to discover these patterns. Some of the methods are sequence based. Then you have structure-based methods, you have interactions-based method, you have expression-based methods, and some of the methods that are sequence-based are based on dynamic programming algorithms. Then you have, as data becomes complex, simple methods may not work, and you would like to apply methods such as genetic algorithms, support vector machines, hidden Markov models. So these are machine learning methods. So there, these methods, once you curate learning data set, they auto learn. Okay, they discover the patterns, and then these methods, when they have learned appropriately, can be used for discovering patterns in the new or the novel data sets. So it may not be possible for me to sort of cover large number of methods in the bioinformatics in this short time. But I would like to give you this simple analogy for knowledge extraction, and text mining is one of the very, very prominent and promising tool for big data analytics. Now I have two paragraphs here. So let us first concentrate on paragraph 1, and maybe you try to read this paragraph. Okay? How many of you are able to read this? Similarly, you have paragraph 2. Okay, so in the first paragraph, you can read it as this message serves to prove how our minds can do amazing things. Isn't that amazing? And isn't that impressive? So here, mind is able to map 7, the number 7 to the alphabet T, and number 1 to alphabet I, and number 5 to alphabet S. So similarities between the alphabet 1 with that of I and 5 that of S has been automatically processed by human brain, and it is wonderful. But look at paragraph 2. Okay, it gives you a different insight. Here, I couldn't believe that I could actually understand what I'm reading. The phenomenal power of the human mind, according to research at Cambridge University. It doesn't matter in what order the letters in a word are. The only important thing is that the first and the last 3 letters be in the right place. So amazing. So our mind has processed this raw data and then is able to make sense out of this data. Now just imagine that you write down all the steps
tacit or non-tacit, explicit and implicit, that have happened in the in your brain. And why? Because then you're trying to simulate how your brain is working for text mining. And if you try to code it in terms of an algorithm to read these two paragraphs, that is how you develop algorithms in text mining. It's really amazing that you need different algorithm, different steps, different processes to read this paragraph versus this paragraph. And without giving any instructions, without feeding in any algorithm in your brain is able to read this. But if you want computer to understand this and to read it for you, you will have to develop a code in 100 lines. That is how you need to pay attention to the processes that exactly happen in text mining. So it is all to discover a pattern at different levels of complexity. So sequences, in particular protein sequences, are full of these pattern NXT. N is ASN amino acid, T is THR, and X is any amino acid. But whenever this tripeptide occurs in a protein sequence, that's a potential glycosylation site. So manually you can search for NXTs. You can also use Control F command. You may not be able to use Control F because X is any amino acid. So any of the 20 amino acid can occur at the place of X. So you need some kind of algorithm, some kind of a program, some kind of text mining. Then there are patterns in the 3D structure. So alpha helices, beta sheets, they come together to form supersecondary structures. There can be tertiary structures, quaternary structures, and whatnot. Similarly, there are patterns in the genomes, there are patterns in the expression profile, and therefore, based on your data to be analyzed and your objective of analysis, there are large number of tools that are offered in the field of bioinformatics. Again, we are not going to get into details of these tools except a few in the sequence domain, and let us look at that. And why I say that let us look at the tools in the domains of sequences, because so what you see as an illustration is a phylogenetic tree of 3 rhinoviruses. Rhinoviruses are common cold viruses, rhinovirus A, B, and C. And this is a depiction that has come out of our own. Nothing in biology makes sense except in the light of evolution. While analyzing the sequences together, you have multiple sequence alignment. Then you have the alignment outcomes. So they help you to identify exact matches, and we call them as identity. Usually identity and similarity are expressed in terms of percentage. Similarities are identical residues plus those residues or bases which have similar biochemical function. Again, we are going to look at it in terms of an example, but alignment also help us to identify insertions and deletions. So these insertions or deletions are inscribed, are shown, regions shown with insertions and deletions. Alignment as a wonderful organizing tool helps us to understand what might have happened in the process of evolution. A region got deleted from one sequence, or a region got inserted in another sequence. So we are going to again look at it with a small example. And this whole purpose of sequence alignment is to study homology, and homology is nothing but evolutionary relationship. So you have orthologs, same genes or same proteins in different organisms. Hemoglobin alpha in human and hemoglobin alpha in horse, they are orthologs of each other. Then you have paralogs. Paralogs arrive due to the gene duplication. So hemoglobin alpha and beta genes in the same host, that either human or the horse, are paralogs of each other. And xenologs are where rare events to prove where these are the regions which are evolutionary related and which have been incorporated due to the process of horizontal transfer, okay, because of the coexistence of a parasite with a host and so on and so forth. Now for sequence alignment, we have separate dedicated courses, and it may not be possible to actually teach you sequence alignment in 5 minutes. But nevertheless, I would make an attempt that, let's say that you have 2 sequences and you are trying to align. So this is sequence 1 and this is sequence 2. Since these sequences are edited for the demonstration purpose.
I'm sorry to interrupt you,
Sure.
madam. We are not able to see your moving slides, madam. We are just seeing your first page only. That is the
Is it?
introduction. Yes, yes.
Oh, I'm sorry. So you should have told me.
Yeah. Thank you.
That's terrible.
Yeah.
Can you see?
Yes, yes, yes. Now it is on slide number 20 or 2.
Oh, I'm so sorry. So this is terrible. Why this could have happened?
It would be better, madam, if you put this on slideshow presentation, so it is automatically moving.
Yeah, that is what I am doing, and this is
Yes,
not the first time I am doing it. Is it visible now?
Yes, yes, yes. It is visible. Yes, it is visible.
Can you see different slides here?
Just move, madam.
I am moving. I am at the first
Now
slide.
this is the 22nd number slide, madam. Pairwise sequence alignments and concepts.
But this is strange.
Yes, yes. Now it is what is bioinformatics. This is the
Right. So can you see now?
yes, yes. It is moving, madam.
Huh?
Yes, ma'am, yes.
So just for the sake of completion, I mean, I was talking, and then you were. I'm so sorry. Somebody tried to stop me, but then I thought that I was talking with respect to first slide, some slide for 5 minutes, and I misunderstood it that the slide is not moving. So this was my first slide. Second slide, can you see?
Yes, madam. Bioinformatics database basis.
No. So second slide. This is second slide.
This is 14. Yeah, this is second slide.
Okay, so I'm able to move slides now only in this mode.
Okay.
All right.
That's
So is full slide visible, or at the side you are also seeing the list of
yes, yes,
slides?
at the side. Yeah, at the side we are seeing.
Also seeing the list. All right. So let us quickly go through these slides. So this is what I had for the second slide, and this is what I tried to explain to you as a discovery framework. I'm so sorry. If I can really, I don't know how to compensate for this loss now. So this was the illustration of gapping void that I showed, and this was the slide where I was trying to explain
Yes.
from population to the atoms, and then I explained to you about bioinformatics being a data-driven approach using this slide. This is terrible and unacceptable. I don't know why it has happened. And then I said that you have bioinformatics breaches. I wish I can just go back and do entire presentation. If you have any free time later on, you let me know. Okay. So this is where I explained that we have data of different types, and I talked about this whole omics series. That is different types of databases, data types available, and then I explained to you how bioinformatics evolved, and then I defined what bioinformatics is, and then how it is a multidisciplinary science, then databases and how data is organized. It's strange. I'm still not able to come to terms with this. And then I talked about these two paragraphs, and I tried to read them for you. Now probably you will be able to make sense. So is it exactly 17th slide that you are seeing?
Yes, madam. It is exactly.
Okay. So these were the paragraphs that I was reading for you. I'm sorry, we have missed on so much of fun as part of this, computational geek that slides were frozen. Extremely sorry about this. And then we are talking about, this is what I was explaining, that it makes sense, and then you have sequence alignment concept. So shall we do one thing? I will quickly unshare and share it again, okay?
Yes.
So that I can make it slideshow, and also but
Whatever you are delivering, madam, as of now, we are already listened properly. No problem. With one request, after the presentation, if you could share this slide on to us so that we can again understand.
what I can do is I can just re-record it with slides and share.
Send this presentation to our committee in charges, so they will share with us. So we can
Is this meeting being recorded?
Yes, ma'am.
Okay. Okay. So hereafter, at least you will be able to see the slides with vis-a-vis with the recording. But let me just unshare it for a minute. Stop presenting and let me presenting once again. So we are at 21st slide and in the slideshow mode, right? Hello? Hello?
No, ma'am, not at slideshow.
Not yet slideshow?
No.
So it says Meet is sharing a window, okay, I will then share my possibly
Your window itself slideshow, ma'am, and just get it back to that window to work out.
No, but this is how I have been doing for how many years and for how many times now.
At times it gets stuck, ma'am, like that.
Huh?
At times it gets stuck like that.
So is it moving now?
No, it is still the sidebar scene.
Oh God.
It doesn't matter, ma'am. We can read it, so it's okay.
No, I can stop and share my screen. Okay, I will. This is least. I don't like to do that, but then I'm sharing my entire screen now. So is it now visible?
Yes. It
And
worked
they are
out.
moving also, right?
Yes,
Yeah,
ma'am.
yes.
Absolutely.
Okay, I'm so sorry for this. All right. So sequence. It is visible. Now I am on 22nd slide.
You're right.
Yes, ma'am.
Right.
Yes.
Oh, you could have stopped me and said it explicitly. I mistook it for, long time I was explaining one particular slide. So I thought, all right. So, here there are two small sequences, okay? And they're edited for demo purpose. So we manually, even if you look at them, you understand there are some amino acids that are common between the two. And you don't need a fancy algorithm to align these, and you can simply, capture the identities that exist between these two sequences towards the N terminal. But then you are not able to capture these identities towards the C terminal because they are not aligning. So if you slide your second sequence just by one position, you can capture the identities that exist towards the C terminal, middle and C terminal. But as biologists, you know that none of these two alignments are actually the optimal or the best alignment solution because they are the linear alignments. So what would be the best solution to align these two sequences? This is the best solution, optimal alignment, where every identity between the two sequences is marked. Okay. So you are able to capture all identities from N terminal to C terminal, which are shown with these pipes here. And there is one small insertion here. So there is so at times when you are analyzing the sequences from the database, you don't know their evolutionary histories. You do not know whether sequence 2 arrived from the sequence 1 or sequence 1 arrived from the 2. Okay, so you are going to interpret the results of alignment in such a way that so many positions amino acid remain the same, they have not changed. There are so many positions where there are substitution, like this D is substituted with C. So these are called as mutations. So there are mutations in some positions, and then there is this position where if we say that second sequence arrived from the first sequence, then we say that there is a deletion of K at this position, or insertion of K at this particular position. So insertion of one break has helped us to maximize the identities between two. So sequence alignment can be redefined as optimization algorithms, which allow you to these substitutions, insertions, and deletions. So so physically this break is not present in this sequence, but sequence alignment help us with simulation process, help us to simulate the evolution. So what might have happened in the process of evolution. And sequence alignments are of two types, as I mentioned to you. You have global alignments where sequences are aligned from end to end, and there are local alignments where some regions in the sequences are aligned locally. Okay, so that's the objective. And then you have these group of algorithms called as dynamic programming algorithm, and there is a very rigorous statistics and mathematics behind all these algorithms. So it may not be possible, but if there is, you try to learn and explore sequence alignment either using the documentation of the global and local alignment programs, or using the original research papers, or we can meet once again to discuss these algorithms in detail. But what I would like to share with you is that when I learned bioinformatics, there were no teachers, there were no textbooks, there were no formal training programs, but I went on along with my colleagues, went on sort of exploring these individual algorithms in great degree of details myself. It took a little more time to understand, read the research paper, carry out that analysis, use the program, do several rounds of back and forth, learning. But at the end of the day, you have cracked the algorithm. So per day, if you sit, then 6-7 hours are enough for you to crack individual algorithm. These days, this algorithm comes with excellent documentation. They come with FAQs, how to use, how to input data, how to generate output, how to interpret the results. Because it is very simple for somebody to copy cut paste sequences and have the sequences aligned. But once you copy cut paste the sequences and hit on the submit button, you should know what kind of computation actually happens behind the screen before you really analyze the results. There is another very, very, very popular program that I'm sure at least a few of you might have heard or used, and the name of the program is BLAST. And BLAST has become so popular that it is like our students use it as a verb, say that I blasted a sequence against the database. So BLAST stands for Basic Local Alignment Search Tool. So here this tool was developed. So if you go on pairwise aligning the sequences with several 100,000 sequences in the database, it would take you eternity to analyze your data of interest. So what is very important is that here you need an algorithm which is quick. It is all right to be approximate, but quick, simple algorithm, that can fetch a set of entries from the database which are similar to the sequence at hand. So you give one protein sequence or one nucleotide sequence as a query, 1 minute. As a query, and then again it's a very famous algorithm which breaks down this sequence in the overlapping words of length 3, and these small words are used to compare with the 700,000 words generated, a dictionary of several 100,000 words generated out of several 100,000 sequences that reside in the database, and then this step is explained here diagrammatically. And this list of words is matched against entries, and then some words which have meaningful scores, good scores, are listed, and then the sequence alignment, this local alignment, is extended in both the directions in order to align the set of sequences. So it is wonderful. Then what is it is most commonly used tool. It has excellent statistical framework, and it also provides you with the statistics that what is the sequence alignment coverage, what is the percent identity, what is percent similarity. So you understand what is extent of similarity between sequences, but this tool also gives you a parameter or a value called as e-value. So e-value is an expectation value. What is the expectation? That the high scoring sequence has been retrieved just due to random chance, just because of bias, composition bias of 20 amino acid, and there is no true biological significant similarity, which I have tried to explore. When you have thousands of sequences and you are searching, challenge is to identify false positive. And here there are a group of owlets, and then there is this little kitty here. So if you look at the eyes and the overall shape of the face and the body, then looks like owl, but then it can be differentiated on the basis of nose and ears visually. So something similar to that. So similar features might contribute to this false positive result, but when you pay an attention, you understand that this is not a true positive. This is definitely a stranger in this group of owlets, and that is what e-value helps you to identify. All right, you have searched the database and identified a set of sequences, and then you would like to align many sequences together, and the tool that allows you to carry out alignment of many sequences is called multiple sequence alignment. Again, this is a very brief diagrammatic representation that you start with 4 sequences, align them in the pairwise fashion. So basically you have n into n minus 1 by 2 possible pair combinations. So you align them, and then cluster this set of sequences together in the order of similarity, so that you are able to generate the multiple sequence alignment. So once you have the order of alignment, so it says that S2 and S4 are very similar. They are the most similar pair, and followed by S1 and S3, and these in turn are very closer to this. So you align S2, S4, insert some gaps using this mathematical model and rigorous alignment. Similarly, you align S1 and S3, and then you align the set of sequences. And then you are aligning this alignment of S1 and S4 with the alignment of S1 and S3 to generate this multiple sequence alignment output. And here you are preserving the gaps that have been inserted in these 2 basic alignments. So this is a very, very simplified version of how multiple sequence alignment look. And this is the output of multiple sequence alignment. It is hemoglobin sequences from human, chimpanzees, goat, pig, horse, and mouse being shown here. So there are multiple sequence alignment output, every line comes from one organism, and then so there are these 6 organisms, but here you are reading the output in terms of columns, okay? And you will see that there are some columns in which amino acid is exactly identical. So for example, here in the 3rd column, all the sequences have leucine, okay? And it has been designated with a star. Similarly, at the second position, you have serine, and it has been designated as star. But then this position, the fifth position, okay, you have methionine in one place, and then THR in other place. So methionine and threonine, okay, it's a favorable substitution, so it has been designated by a colon, and then so very high similarity is designated by colon, and the similarity of next level is designated with a dot. So this is how, and when you see these stretches of stars, so sometimes this peptide can be a consensus peptide representing this group of organisms. So consider an experiment of aligning all hemoglobins in the world from C. lamprey, the lowest organism, to human, and if you see a stretch of stars, then that particular stretch serves as a motif, serves as a signature for this group of proteins. So that's the significance of multiple sequence alignment. But then, once you have had multiple sequence alignment, you would ask this question: how similar is similar and how different is different? So, the purpose of how similar is similar and different is different depends on the question you want to answer using alignment. For an evolutionary biologist, sequence similarity more than 30% is good enough to establish the theory of evolution. For a biochemist who's trying to validate the function of the protein, 35 to 40% similarity is good enough, provided the residues of the active site are exactly identical. If there are substitutions in the active site residue, you will detect, or it will reflect in the readings of the experiments that you are performing. For a molecular modeler who's trying to predict three-dimensional structure of the protein, again 30% of identity and 40% of similarity is good enough. For a drug developer, same is the case, but think of immunologist. Okay, so at times, for example, SARS-CoV-2, spike protein of different lineages of SARS-CoV-2 has 95% of identity and 97% of similarity, despite there are varying levels of vaccine efficacy. So these individual mutations are very, very important. Another example from the perspective of immunology that I can give, let us say that you align human and chimpanzee hemoglobin, 98% of identity, 99% of similarity, but you inject human hemoglobin in chimpanzee and chimpanzee hemoglobin in human, both the hosts will identify the non-self nature of the molecule injected and will be able to mount the immune response against that. So how similar is similar and how different is different depends on the purpose of objective. So, pairwise alignment, you establish homology. Multiple sequence alignment, you use to discover the patterns. Then you have phylogeny as a set of analysis, which, help you to draw the phylogenetic trees. And they have become an integral part of, bioinformatics analysis. As biologists, you may have a gene or protein sequence from a group of organisms, and you would like to see or you would like to build the evolutionary history for the set of, organisms and the sequences you have. Again, molecular phylogenetics is a branch in bioinformatics, where it may not be possible to introduce you with various methods in detail, but I can help you understand the overview that there are alignment-based methods, and the alignment-based methods are classified into two categories: distance-based and character-based, and then there are Bayesian-based methods, and they can be used. There are various applications of phylogenetic analysis. You can look at evolution of genes, evolution of species. You can, let us say that you are a botanist and you're trying to, you come across a species of a new grass, it was discovered a few years ago. So you want to establish whether it is a new species from the Western Ghat or a variant of the known one, you can have the characteristic gene sequences obtained in the wet lab, carry out this phylogenetic analysis, and on the basis of the placement of this operational taxonomic or the taxa unit, you can talk whether it is a new species or not, or you can use it to differentiate drug resistance, multi-drug resistance TB from the normal TB isolates. So there are variety of applications, and not necessarily phylogenetic analysis, but for several other purposes, calculation of distance between sequences is very important, and it is very simple. So you align the two sequences. Okay, here again they are edited for demo. So sequence 1 and 2, okay, they are exactly identical except for these two positions. So that means there is 80% of similarity. Okay, so distance is usually 100 minus 80, so 20% of distance, but it is usually referred in the scale of 0 to 1. So 1 minus 0.8 is 0.2% distance. So S1 and S2 has 0.2% of distance. So it is simple distance calculation formula that can be given as an input to a program such as neighbor joining method, where it starts with a star-like topology. Let us say that you have these 8 sequences at 8 entities at hand, and you are trying to generate a phylogenetic tree. Then the most similar pair is identified first, and a common hidden evolutionary ancestor is assigned to that, and that evolutionary ancestor is linked to the centroid to which all other sequences are at equidistance, and you go on iteratively carrying out this procedure until you have generated a phylogenetic tree. So usually you have rooted versus unrooted trees. So this is an unrooted tree where there is no specific root. But rooted tree is you have an ancestral sequence from which all descendants are shown. I have not included it in this purpose. In view of our time, it may not be possible to go through these character-based or parsimony method. But again, you start with the multiple sequence alignment, and then you generate various tree topologies. Okay, based on the sites under selection, and these dots indicate number of mutation along this site, and based on that you can come up with multiple tree topologies, and you prioritize or you take the topology where you can explain data with minimum transformations or minimum changes. For example, this particular tree topology says that third and second are closest and first and fourth are closest, but these transformations can be explained using 4 dots, whereas here it can be explained using 3 dots, and here also 3 dots. As against, for this particular position has least information contained. But if you look at the position 5 in multiple sequence alignment, just with 1 dot, it is able to build a tree topology that it says that 1 and 2nd are closely related, and 3 and 4 are closely related. So let us go back to MSA and look at this site 5. So this 1, 2, 3, 4, and 5. So this is an informative variable site. So when you look at this, these 4 sequences can be quickly divided into 2 groups on the basis of 2 Gs present here and 2 As present here. And then if these 2 sequences have had a common ancestor, and these 2 sequences had common ancestor, their ancestral sequences require just 1 change from A to G in order to interchange these branches. And this is the basic principle of maximum parsimony or minimum evolution method that we talk about for phylogenetic analysis. So usually, we have been working in the field of viruses, and we have been using these phylogeny methods, but soon there are more than, scientists world over started with 150 vaccine candidates, and then you have several companies in India which are coming up with vaccines, and you are aware, and I'm sure as teachers and thought leaders, you are tracking, how exactly different vaccines are performing, and of course, the equitable distribution of vaccine is a challenge, but nevertheless, we need to vaccinate a large number of population, student population, before they can come, and herd immunity can really be generated. So, so bioinformatics, a specialized branch is immunoinformatics and vaccine design, where you can start with genomic sequence of the pathogen and can come up with using reverse vaccinology, identify the antigenic components, and then use or apply structure-based vaccinology to come up with candidate vaccine. And this is what a few of the scientists have done in this particular phase. And some of the vaccine candidates are in the clinical trials. Some have been approved, and we expect a few more candidates to come up. So all said and done, to summarize, whether it is basic research or translation research, every experiments begin in silico, and bioinformatics at the core. So you start typically with the reference or literature searches, then look at disease databases, look at genome databases, look at the corresponding proteins, carry out some sequence analysis, map those interesting variations on 3D structures, study domain interactions, and all these basic analysis can be used for the translation purpose to describe development. Yes. So your gateway for bioinformatics begins at NCBI. So this is the URL. Please feel free. There is wonderful documentation, including some of the tutorials available. EBI is another major bioinformatics resource that you can use, and Expercy at Swiss Bioinformatics Institute. Okay, this is the third major resource. So you can use these resources for bioinformatics training for yourself or for your students. This is PDB, which archives protein data bank. Just for 10 minutes, we have this sneak peek into history and beginning of bioinformatics. It's Margaret Dayhoff who has been responsible, who's considered as pioneer for the field of bioinformatics. She was doing her PhD in early late 60s, early 70s, and then her postdoc. And that is where biological data was coming. Sanger sequencing method was there. Computers were making rounds, and that is how she merged biology with computers. And then on the other hand, you have heard about Professor G.N. Ramachandran, who talked about reduction of three-dimensional data in terms of 2D plot, Ramachandran plot, that all of us use in order to validate quality of protein structure. So it is an example of data compaction. So these two can be credited. Bioinformatics in India came up as a BTIS network, and we have these centers distributed all over India. We at Bioinformatics Center at ISPPU, we have R&D programs, we have HRD programs, and our thrust area is virus bioinformatics. So these are the different disciplines in which my colleagues are working, and they are sub-disciplines of bioinformatics. So we have several courses designed around these major disciplines, and that our MSc Bioinformatics and PhD Bioinformatics programs are tailored around that. We use different analytical pipelines, and we curate data, develop databases, we develop algorithms, we develop servers and several applications. So these are some of the snapshots of the servers which have been developed at our center, and they are available in the public domain. These are examples of some of the databases that we as a faculty have developed. Because virus bioinformatics is our focus, we also happen to work on SARS-CoV-2 right from the beginning of the small outbreak in Wuhan. So first sequence of SARS-CoV-2 came in second week of Jan, and ever since then we were working with these genomic sequences and genomic sequences of other beta coronaviruses, and that is where this is one of the first few papers from India in April that we came up with this this explanation that how SARS-CoV-2 has learned to jump the species, okay, from bats to intermediate host to human. And then as part of my editorial assignment at Infection Genetics and Evolution, we as editors have reviewed the progress of SARS-CoV-2, how mutations have made the difference in the span of an year, and we wrote this review manuscript in the journal. So for you, in order to motivate youngsters, yeah. So Delta emerged in India in September last year, okay, and it has been circulating. So we are analyzing, in fact, all circulating VOIs and VOCs in India using Indian genomic data. We'll soon be writing that paper. There are papers coming up which talk about, how effective the vaccines are with respect to emerging lineage. So first and foremost, and the bottom line is, vaccination with available vaccine is a must. Vaccines are useful because they help us generate immunity against the SARS-CoV-2 virus. Viruses, being RNA viruses, are amenable for large number of mutations, and viruses will continue to mutate. New lineages, VOCs and VOIs would emerge, but broad level immunity is conferred by most of the vaccines which are given in all the countries. So vaccination is a must. So there are also studies going on with respect to breakout infections. That is, somebody has already had 1 dose or 2 dose of vaccines, and he or she encountered
Good afternoon, Bhange sir. Good afternoon.
Good afternoon, sir.
Good afternoon, Rasade sir. Good afternoon.
sir.
थांबूया अजून लोक बहुतेक लंचला आहेत.
दोन मिनिटं.
नो सर, नो प्रॉब्लेम सर. या सर. भांगे सरांचं अभिनंदन.
हा, डॉक्टर झाले.
तर त्यांचा आवाज येत नाहीये पण.
त्यांनी म्यूट केले.
अच्छा, पवार सर येऊन बसले काय मागे?
पवार सर आहेत.
दिसत नाहीयेत.
काय आहे?
नमस्कार सर, नमस्कार.
आहेत पवार सर.
आवाज येत नाही. माईक भांगे सर.
त्यांना माईक,
भांगे सरांचा माईकच चालू नाहीये.
नाही, त्यांनी माईक बंद केला त्यांचं.
अच्छा,
त्यांना ऑपरेटर करायचं ना.
ओके. थांबूया एक 5 मिनिटं अजून. लोक बहुतेक
आज दुपारी सर सेशन असं टेक्निकल सेशन.
बर.
डिपार्टमेंटचं एनिमल दाखवणार आपण, सीआयएफचं.
बर.
त्यांचं डेमॉन्स्ट्रेशन, सॅम्पल प्रिपरेशन वगैरे, सगळं अनालिसिस वगैरे.
पण मला वाटतं ते HPLCचं काहीतरी होतं ना. मला वाटतं
नाही, ते होतं HPLCचं. पण काल कुंभारचा फोन आला की आपण आमचं दाखवूया, सीआयएफचं दाखवूया म्हणे.
ओके.
तिकडे एनएमआरचं आहे.
नाही, चांगलं आहे. उलट ते जास्त चांगलं आहे, कारण लोकांना हे करायला. नमस्कार शिंदे सर. नमस्कार.
त्यांना माहिती होईल लोकांना. बाकी
हो,
विद्यापीठाचं माहिती होईल, फॅसिलिटी सगळ्या.
शिंदे सर नमस्कार. शिंदे सर, आवाज
नाही, म्यूट आहे.
नमस्कार
सर. माझं NMRचं दुपारी प्रेझेंटेशन आहे. NMRचं.
हो, डेमॉन्स्ट्रेशन दाखवा म्हणते.
बर. NMR आणि पूर्ण अनालिसिस सॅम्पल पूर्ण अनालिसिस
सॅम्पल प्रिपरेशन, अनालिसिस आणि इंटरप्रिटेशन दाखवा.
आम्ही मागच्या वेळी 5 दिवसाचा कोर्स घेतला, सर्टिफिकेट कोर्स. आणि जसं आता ते शिंदे सरांचं लिहिलं होतं बघा, HPLC डेमॉन्स्ट्रेशन, तसं आम्ही आमच्या इन्स्ट्रुमेंटचं DST फीस त्या अंडर जेवढी इन्स्ट्रुमेंट झाली त्याच्यातलं UVचं डेमॉन्स्ट्रेशन दिलं. त्याच्यानंतर GCचं लाईव्ह डेमॉन्स्ट्रेशन दिलं सर, म्हणजे पूर्ण अनालिसिस सॅम्पल. त्याच्यानंतर HPLCचं डेमॉन्स्ट्रेशन दिलं आणि IRचं पण डेमॉन्स्ट्रेशन दिलं. म्हणजे आम्ही सगळं 4ही इन्स्ट्रुमेंट आमची व्यवस्थित एकदम चालू आहेत. त्याचं सुरू
मागच्या वर्षी केलं होतं सर. ट्रेनिंग घेतलं होतं.
बर.
HPLC दाखवलं, ASTGA.
बर.
इन्स्ट्रुमेंट दाखवलं होतं डेमॉन्स्ट्रेशन. आणि मागच्या वर्षी वर्कशॉप घेतलं होतं.
पण आता आमचं सेट झालंय आता सगळं. आणि आमच्याकडे आता GC, कॅपिलरी GC, त्याच्यानंतर ॲजिलंटचा आहे. HPLC शिमाडजूचा आलाय चांगला. आणि त्याच्यात दोन्ही फॅसिलिटी आहेत सर, UV डिटेक्टर आणि RID डिटेक्टर. दोन्ही फॅसिलिटी आहेत. त्याच्यानंतर 150 सॅम्पल, 256 सॅम्पल्स अनालिसिस 1 स्ट्रोक सुरू करू शकता ऑनलाईन. हो. आणि IR ब्रोकरचा टेन्सर 2चा आहे. टेन्सर 2चा म्हणजे विद्यापीठातला जो आहे तसाच. आणि ATR प्लस KBR पेलेटची फॅसिलिटी पण आहे. आणि रेंज जी आहे सर ती सगळ्यात महत्वाची 400 पासूनची आहे. सो दॅट इज मोर इम्पॉर्टंट. आणि UVDRS आता आमच्याकडे तो पण आमचा चालू झाला. त्यामुळे एकदम आमची लॅब आता एकदम परफेक्ट सेट झाली सर.
नाही, तुमच्या का आता सॅम्पल घेऊन यायला पाहिजे
सर. अगदी या आता तुम्ही कधी पण येऊ शकता. आणि GC तर एकदम रोबस्ट आहे एकदम. आणि वी हॅव सेट इट व्हेरी नाईसली. तुम्ही खरं ऍक्च्युली तुमची लोकांची सगळी गरज आहे. आम्हाला यायला पाहिजे. तुम्ही आम्हाला सजेशन्स द्यायला पाहिजेत, कारण तुम्ही आमच्या अगोदर हे सगळं सुरू केलंय ना ऍक्च्युली. आता म्हणजे
आता आमचे इन्स्ट्रुमेंट बंद पडायला लागले हळूहळू सर.
अच्छा. नाही, पण आम्ही आता तुमच्याकडे बघूनच आम्ही ते आमचं सेट केलंय. कारण आता तुमच्याकडे कसं CHA आणि अनालायझर आहे ते युनिक आहे ऍक्च्युली. असं म्हणजे ते फारच महत्वाचं आहे ते. आणि असं
ठीक आहे, सुरू करा सर. लोक आलेत.
चालेल.
या
22 लोक आलेत.
Now we are on the second last day and second last session of our today's speaker, Dr. Sachin Sampatrao Sakte from Modern College, Shivajinagar. So, sir had completed BSc and MSc from University of Pune and completed PhD in 2019 under Dr. C.V. Rode from NCL on solid acid catalyst for carbon-carbon bond formation and CO bond cleavage in organic transformation. Sir also qualified GATE in 2002, NET and SET also. Sir has experience of teaching as well as industry. Sir worked as a lecturer in Government of Maharashtra Institute of Science, Nagpur, and also sir worked as a lecturer in Ismail Yusuf College, Jogeshwari, Mumbai, and also work as assistant professor at present there in the Progressive Education Society, Modern College, Shivajinagar. Sir also work in industry, that is in Sudarshan Chemicals Industry, Pune, for about two years. Sir has published about 16 papers in reputed journals. The important thing is that sir has experience, that is experimental skills sir have, that is synthesis of heterogeneous catalyst, especially solid acid using heteropolyacid as well as ionic liquid, and its application in chemical transformation for fine chemical synthesis and synthetic methodologies. Sir has carrying high pressure reactions in batch as well as in continuous reactors, isolation of compounds by column chromatography, and structural isolation by using IR, NMR, HRMS, mass fragment pattern. Sir has also experience about handling HPLC, GCMS, X-ray powder diffraction, then micromeritics for BT surface area, TPR, TPO, ammonia TPD, as well as FTIR, as well as Pyridine IR instrument. Sir has also computer skills like ChemBio draw, office draw for chemical structure and interpretation, origin to plot graph, literature survey by using CA and CD, then SciFinder and other using that how to find literature review. Sir has completed 2 minor research projects which are funded by UGC as well as Savitri Phule University. Sir also have experience about stage research, early stage research on NARI-LAR project entitled Fluid Thermophysical Properties, Transport Properties of Ionic Liquids with Other Co-solvents in Ionic Liquid System under the Marie Curie program in the Spain. And under their guidance, about 25 students has completed their project of MSc as well as some research students. So with this short introduction, I will welcome Dr. Sachin Sakte on behalf of Professor Ramakrishna Memorial College and this FDP program on techniques for research in chemical and biological science. So we're going to talk on the very new technique which is used in recently, that is a Pyridine IR spectroscopy. What is use of this Pyridine IR spectroscopy? Because in recent papers, to get a high impact factor, this technique is very essential and they require about this Pyridine IR spectroscopy. This technique is used for both chemical and biological samples, and about whether it's acidic or basic nature may be there that we can find out. So without taking not more time here, I request Dr. Sachin Sakte sir to deliver talk. Thank you, sir.
Thank you, sir. Thank you. Am I audible? Am I audible?
Yes, yes.
Yeah. Sir, thank you very much, sir. Dr. Rasade, sir, for giving this very sweet and very good introduction of mine. But I feel I am the youngest one to speak in this 5-day international program, because I have seen the list of the speakers who are well-known and renowned scientists. I also thank all the organizers of the Ramakrishna More College who had given me this opportunity to share my work.
Sir, please unmute, sir.
Sorry for the internet disconnection, but I have a good collaboration with all the faculty members of the Ramakrishna More College, and I have a few very good publications with Pawar sir, and we had a good work done together. So I will be putting this some new techniques of the characterization of the heterogeneous catalysis, which we have already performed in the our research. So I thank you all for giving me this opportunity once again, and I would like to start with sharing my slides. So with the permission of the chair or with the permission of the organizers, I would like to share my slides. Yeah, I think CO bond and the fingerprint region, 400 centimeter inverse. Double bond, everybody is knowing, coming around C double. Alkynes and nitriles are in the region that is for the triple bond, and what will happen if it is a dimer, if it is an oligomer, we can have the broad spectrum analyzed and we can come to the final conclusion with the bands which are observed in the infrared spectra that we are having. Yeah, and we are having the hello. I'm saying some. I'm listening some voice. Yeah. Okay. Any question? If you have, you can ask me. Okay.
Okay.
Yeah.
So, Sakte sir.
Yeah.
Go ahead. No
yeah. Okay. So this is about the ring strain. Actually, we can monitor one of the very good application of the infrared spectrum, that we can have the ring strains. Also, we can identify hydrogen bonding. We can identify symmetrical stretching, or in the symmetrical compound. We can identify with the help of the various frequencies which we are observing in the infrared spectrum. That if you observe the 3-membered, 4-membered, 5-membered, 6-membered, and 7-membered carbonyl compounds, you will observe that because of the ring strain, the frequency is going on increasing as the ring size goes on reducing. These are the applications which we are coming to the conclusion with the study of the various organic compounds, actually. And we are actually still we are talking about organic compound, only organic compound. So why I am specifying this? Because everybody is very well familiar. Everybody is very familiar with the organic compound and infrared spectrophotometer. Majority of the researchers are also familiar with the only the technique which is used for the organic chemistry. But Now, why we are talking about IR so much in the inorganic chemistry? So inorganic chemistry, or you can see the catalysis, or the very different branch of the chemistry, that is the catalysis. So catalysis, everybody knows why we are doing it. So biomass conversion is there, a very basic and very vast area. Biomass conversion, value added chemicals, like we are in the drug synthesis or in the additives also, like then fuels. Catalysis is used in the fuels manufacture. Sustainability, the major issue or the crisis, CO2 fixation. Apart from this, all organic chemists are working in the various organic reactions like condensation, addition, substitution, rearrangement, coupling, photochemical, multi-component synthesis, substitution reactions, alkylations, acylations, protection, deprotection. So all these reactions are basically because of some active species, actually. So whenever we are doing the catalysis, whenever we are doing the any organic reaction, we need some. Either we are doing it by using the acid, either we are doing it by using the base, either we are doing it by Lewis acid, by using the various reagents. Finally, it comes to the acidity and basicity, actually, or whatever we are proposed for the catalyst, for preparation of the catalyst, is nothing but the acids or bases, which we are actually using for the majority. I am not saying all, for the majority of the organic reaction. So generally, if you see, the Lewis acids are generally, I had given a few examples of Lewis acid. Aluminium chloride, FeCl3, ruthenium chloride is quite costly. So we can say, but ruthenium chloride is also used nowadays in the various organic reactions. Indium chloride is there, indium bromide is there. Triflets are the best used material in the various organic synthesis. One of the major branch which is coming out in a very fantastic way, that is the Bronsted solid acids. So solid acids concepts itself is a very interesting thing, is that solid acids like phosphotungustic acid, like phosphomolybdic acid, we have used it actually, but we have not thought of using this as a catalyst. And I have, in my work also, I have used it. My group, the whole group in the NCL was working on this solid acid catalyst. And for various organic reactions like CC bond formation, CO bond cleavage, CN bond formation, so many things we are doing with this Bronsted acid catalyst. And this why we need this? Actually, these are the polyoxometallists, and the major inorganic chemists may be explaining this very vastly, but I would like to tell you that this is having a very high molecular weight. Firstly, okay, it is a super acid. Actually, it is easily available. It is highly pure. It is highly crystalline. See, what are the properties? See, it is easily available. It is high molecular weight. You require a few grams, few milligrams, rather 1 to 2 milligram of this compound is also sufficient to do a lot of things. Brilliant chemistry can be generated with the help of this. So these Bronsted acids are also there, and this acids are having only one drawback. Actually, they are having a very small surface area. Remember, they are having a very small surface area. So here is the point of our interest actually. So they are having so many properties, but their surface area is very low actually. Okay. So these are the supported or non-supported Bronsted solid acid catalyst. If you see very carefully, HPA, that is the heteropolyacids. That is nothing but phosphotungustic acid, PW12O40. That is phosphorus, tungsten, and oxygen. That is PW12O40 is the compound's name actually. So HPA, this can be supported on the various supports actually. So I am coming to the next point actually. So these are the active species. Now what are the active species? These active species actually The active species in the catalysts are aluminum chloride, iron chloride, indium chloride, or ruthenium. Actual metal is the active species. Or if you are having the Bronsted acids like phosphotungustic acid, phosphomolybdic acid, somebody will say, sir, why not HCl? Of course, we can have the HCl also. You can support HCl also. You can support H2SO4 also. Sometimes somebody will say, why not chlorosulfonic acid? Yes, chlorosulfonic acid also. And we have used it actually. So it is not like that we are restricting ourselves to the particular solid acid. No, but only point is that your support should be that much active, or your support should have that much properties. So generally, what is actually the heterogeneous catalysis designed? How it is designed? Okay, so we are just going to, this is the categorization like Lewis acid, then Bronsted acids, and now we are going to have, this is the supports actually. So generally what is done, the supports are taken, and on that supports we are supporting the active species actually. Now when we are supporting the active species, what is the property? What is the basic property you should have? You should have the nature, that is microporous, mesoporous, or macroporous. There are three categories actually. So microporous, mesoporous, and macroporous. So mesoporous is having a diameter actually, the cavity diameter or the shape. If you see, this is 2 to 50 nanometers. Microporous material, these are the zeolites, actually zeolites. These are the zeolites which we can synthesize in the laboratory also, and they are having the surface, they are having the pore size, cavity size is less than 2 nanometers actually. So this is mesoporous, this mesoporous is having 2 to 50, and macroporous is greater than 50 or below, above 100 nanometer actually. So these are having like an honeycomb structure, you know, see, if you see this, I have shown you the MCM41 actually. So this is an honeycomb like an, it is a cylindrical actually, but it's not looking like this. If you see the TEM image of it at the surface, you will see that a honeycomb sort of thing is observed actually. Okay, so you have a very good support. Now why we need the support? Support is having a various properties. Now these are the examples of the zeolites actually. These are the synthetic zeolites which you can prepare in the laboratory also. I'm going to show you how this can be prepared in the laboratory. This MCM41, MCM48, MCM50, SBA15, SBA16, they are not a codes, remember actually. They are not the coded languages. No, it is a mobile composition of matter. Mobile composition of matter. So it is the mobile composition of my matter is a company which has discovered this actually, and they have given the codes. 41th batch was successful, or some code was given 41, and that's why they named it as MCM40. So this type of things we have to understand, that we have to select a support. Now what is this actually MCM? This MCM is a silica. You will not believe that this is a silica. Why we are calling it as a silica? It's a simple silica. Our laboratory by doing the hydrothermal treatment. Now this is the properties actually. Why we need the support actually? So I will be telling to you, I will be coming to the point of MCM41. Before that, I would like to summarize the properties of the support. Why we need the support? So we can take the various supports like silica, alumina, activated carbon, graphite, Mont clay. Mont clay is having a very good properties in the various organic synthesis. Bentonite clay, it's a pillared clay. MCM41, MCM48, SBA15. What are the properties? The properties are high surface area. High surface area. Can you imagine a material having a surface area about 1400 meters square per gram? And you can design it in a laboratory. Yes, we can design this material in the laboratory, and I am designing in my college actually. I can prepare the material. It doesn't require a very sophisticated instrumentation. Nothing. And we have designed it actually. High dispersion ability. Now here is the point, dispersion ability. Dispersion ability means what? We are actually dispersing our active species on the surface area. So see, if you get the very high surface area, you can disperse your active material on the surface area very uniquely and uniformly. The point is, it is the uniform distribution. It is the uniform distribution of the material. So you can disperse the material, active species. See, the catalyst is active only when it gets dispersed on a support uniformly. Then and then only its tough and turnover frequency and turnover number will be very high. So if you want to increase that, this dispersion is very important. And for that dispersion, you need a very high surface area and a very, very perfect structure, or a structure like MCM41. So it, again, another thing is that the surface itself may have the Bronsted acidity or the Lewis acidity. The surface is already having the Bronsted acidity, like zirconia. If you consider the zirconia is already having the Bronsted acidity. Its catalyst life is increased. Catalyst life, number of cycles you can increase with the help of this type of selection of the support. Hexagonal porous structure, its cavity is increased, your active species fitting is very important. Because actually, remember, we are considering two types of properties: textural properties of the catalyst and the microscopic properties of the catalyst. And when we are doing the characterization, we are doing the characterization of two types. One is the textural one and another one is the microscopic one. So in this case, the hexagonal porous structure is coming under the textural one because it is having a porous structure. Its porous structure is very easy to hold the active species. It gets set in that porous structure, and you are getting a very active catalyst. Mesoporous material. Its mesoporous material means its porosity should be very uniform. Thermal stability. If you have the support, you can increase the reaction temperature to the certain level also. And finally, the recyclability. This all properties comes with the support. You can design a new support. If you have any unique idea, if you can design the support, you can have zinc oxide is very well studied material. Zinc oxide.
रेंज गेली की काय सक्सेस म्हणजे रेंज गेली का
Wait for some while, sir is going to rejoin again.
Okay. फोन कडे तरी सुट्टी झाला ऐक ना काय होतं हा
Okay, dear participant, wait for some moment. There is some technical error. Something like this. फोन करना एक फोन कर फोन रख ले फोन कर Dear participant, on your camera. We take the फोन कर काय नेटचा इश्यू झाला की काय
व्हिडिओ ऑन करा म्हणा फोटो घ्यायचे
Dear participant, on your video camera, we want to take some photographs for attendance purpose. And it is for program schedule. Okay, on your camera. Bhagat madam?
Yes, sir.
On your camera.
Sir, it's on, sir.
It's on?
Yeah.
Okay, okay.
I'm going from two devices.
Kadnande sir. Hi, sir.
बरं सक्ते सरांना लागला का फोन?
हा लाव करतायत रसाळ सर करतायत. उज्ज्वला मॅडम. अविनाश, संतोष राठोड, पटकन करा.
सर माझा कॅमेरा ऑन
आहे का? बरं ठीक आहे. ज्यांचे नसेल त्यांनी करा. भूषण हिरे, भंडारी सर, शरद जगताप
सर ऑन आहे ना सर?
ऑन आहे सर, ऑन आहे.
रमेश सूद, शरद जगतापचा नाही दिसत मला ऑन. रमेश सूदचा
येस सर.
ओके. सोमनाथ वाघमारे.
Yes, sir.
ऑन
Camera is on.
डिस्कनेक्ट
Yes, sir.
होते सर परत.
कसं
Camera is on.
काय परत ऑन करून ठेवा ना, काय अडचण येत नाही.
नाही ऑटोमॅटिक नेट वगैरे झालं ना सर, परत डिस्कनेक्ट.
तो होतोय का? बर. निलिमा देवी मॅडम नाही करत. कॅमेरा ऑन करा.
ऑनच आहे सर.
हा ना, ठीक आहे. ओके. And day by day, number of participant is decreased. I don't know. Okay. 132. मोबाईलचं नीट घेऊन जाणार मोबाईलचं नीट येते इकडं कुठं म्हणजे कॉलेजचं नीट गेले का ते लॅपटॉप आणि एक मोबाईलचं नीट ठेवायचं म्हणायचं मला किती वाढत आपली कॅमेरा ऑन करणार 36 दिसतात का 1 2 3 4 5 6 40 आता जॉईन सत्तेदारांच्या कॉलेजची पॉवर दिलेली आहे त्यामुळे त्यांची कनेक्टिव्हिटी दिलेली आहे ते मोबाईलवर ट्राय करतायत 2-3 मिनिटात ते सुरू करतील Now I giving some instruction to participant. I request all participants submit your assignment on Google Classroom. Don't send on WhatsApp or email of any coordinator, the assistant coordinator or coordinator. But the students, delegates have sent the assignment on my WhatsApp also. So I request don't send the personal WhatsApp or mail of a coordinator or any teacher. We submit assignment on Google Classroom. Upload on Google Classroom. Okay, that assignment only count. Tomorrow we take one test for 50 minutes. Time duration for the 1 hour. Based on the questions, whatever topic delivered by resource person in a FDP. Therefore, 10 questions from botany subject, 10 questions from zoology, and other questions from the chemistry. Because of the depend on the resource person, these are the weightage for the MCQ questions. Okay, I can repeat. Tomorrow we conduct after completion of all 3 sessions test at 2:30 to 3:30. ये 2:30 test 2:30 to 3:30 p.m. Duration for 1 hour and marks for 50. All questions are compulsory. 50 questions are MCQ questions are given for test, which is based on the content whatever delivered by the resource person. Got it? I hope everybody. Another instruction for the participant, we fill out the form, registration form on HRDC website of SPPU. UGC HRDC website. SPPU. कॉलेजचे नेट गेले ते मोबाईलवर करतायत कनेक्ट. आता मोबाईल काय होतं फोन करणार एकदा कोणी केलं This is at 2:30 to 3:30. Tomorrow there is a 3 session. The important sessions are there. First one is Dr. D.D. Dole. It is shortly known as the DDD. Second one is Milind Kulkarni from CIMET, and third is Nagarkar from the there is also from this Amazon. We are having a Limca book record part, that is a resource person. For 4 sessions start sharp at 9:30. It is delivered by the D.D. Dole, Dr. D.D. Dole from Chemistry Department, Emeritus Professor from the Department of Chemistry, Savitribai Phule, Pune University. Second one, this session start from first session is by the Milind Kulkarni, so there is short change. Milind Kulkarni at 9:30. 11 p.m. AM the D.D. and from 1 p.m. is Ankita Nagarkar. And test is 2:30 to feedback. Feedback से वो बोल लो ना सांगितलं Okay, pandemic situation or COVID-19 situation, we are trying to develop new methodology or new option. That is where we conduct the program on virtual mode. When technology giving the cooperation, it is better to conduct the program in online mode is very easy. But sometimes there is a technology issue is arising. We cannot help you for that. Okay, therefore, we are apologize regarding the technology issue arises for the connectivity of the resource person. We are trying to resolve the problem of connectivity, and within 5 to 10, 3 to 4 minutes, he will be join the session.
Sir, test is also for students, or is it for faculty only?
Both.
For both. Okay, thank you.
Yes. Faculty as well as student. Your performance of FDP assess in terms of your attendance, then your assignment as well as your test and feedback, online feedback of every day. This is the parameter to decide your overall form of performance in FDP. Your environment decide the feedback and your attendance, and your presence of mind decide the assignment submission and whatever marks obtained in the test. का बोलतोय आता केला परत फोन काय मला जरा मोबाईलवर नेट नाही का
हॅलो पवार सर. पवार सर. भांगे सर. हॅलो.
हा कोण क्वेश्चन कोणतरी विचारत होता.
हा हा
बोला.
मी बोलतोय सर पवार सर.
बोला ना.
काही पार्टिसिपंट आपल्याकडे असतील त्यांच्याकडे काही फॅसिलिटी चांगल्या उपलब्ध असतील रिसर्च साठी किंवा काही ते सर्व्हिसेस देत असतील तर
येस येस
हा तर फीडबॅक घ्या कोणाचा तरी म्हणजे
चालेल चालेल.
कोणाकडे काही फॅसिलिटी असेल चांगले
रिसर्च
किंवा कोणी काही सर्व्हिसेस
फॅसिलिटी
द्यायचं असेल रिसर्च साठी तर शेअर करू शकता तुम्ही.
Okay, I request you participants or delegates, please share your views regarding any facility, research facility available with your institute. Any research faculty, instrumentation or other facilities is available in your institute or college. Anybody want to share the views or give the information to?
Yeah, sorry for the thing, but now I'm sharing with Wi-Fi on Wi-Fi.
Yes. Sorry. We are waiting. Okay, no problem.
Can you see this now?
Yes.
Can you see the presentation?
Presentation. एक मिनट.
No.
No, we not.
Okay, sir. Okay. Wait. So the total college network has dropped down. So I'm extremely sorry for that, but I can't help it out there. So I have made the arrangement with Wi-Fi. Let us hope it will work. Okay.
Okay, sir. Okay.
Yeah. Maybe the size is the issue, which is not getting shared actually.
Screen share hello. Bhagya sir. Allow them to share the screen.
Yeah, maybe.
Okay, Bhagya sir, allow to view for the share the screen.
Yeah, but still I have shared it, but it is not seen. I have already shared the screen.
Sir, see. First we close your whatever PPT we open, and again you open it and then share it.
I will just do that. Just a minute.
First close the PPT
Yeah, sir. Yeah. Definitely, sir. Just.
Then open it and then share.
Yes, sir. I'm saving it actually, and then I will share it.
Mange sir, come here. हा त्यांना सांगा शेअर कशी करायची
केलंय सर केलंय एक मिनिट सर 1 मिनिट
ओके
या सर एक मिनिट सर 1 मिनिट फक्त ओपन होईपर्यंत जस्ट अ मिनिट
येस
टेक्निकल इश्यू आल्यामुळे मला काहीच करता येईना झालं कारण का अख्ख्या कॉलेजचं नेटवर्क डाऊन झालं त्याच्यामुळं आय एम एक्स्ट्रीमली सॉरी फॉर इट बट I have not permission.
No permission. Just.
It is. Yeah. Now I got it.
Yes.
Yeah. Now it is seen. Sir.
Yes, sir. Yes.
Okay, sir. Just.
Now continue, sir. Continue.
Yes, sir. Just a minute, sir. So I was on the point of discussion about the pyridine IR instrumentation part. Okay, so this is the actual instrumentation which we are using here. Now, if you see very carefully, this is a normal pyridine IR. This is normal IR spectrophotometer, but here there is a praying mantis attachment. Now, this attachment is not coming with the pyridine IR. Let me clear you very seriously and very frankly. This is a praying mantis which costs round about 15 lakhs rupees extra, other than the IR instrument actually. And that's why this is so important and so essential, you can say, because it is instrumentation is designed as per our requirement. Because the designing is coming later. First our necessity was considered and then the instrument was designed, and these are very specialized techniques done by the Perkin, Elmer and Brooker only. Remember, you will not get the praying mantis with every IR because they will not be in a position to give you the praying mantis. This is the third party attachment. Remember once again. And those who want the information of purchasing this type of thing, just contact me freely. I will be giving you the total details about it because I have been the part of purchase, installation, commissioning, training, everything I have done it. Okay. Now, if you see, this is a dome. Actually, it is a dome. This is a KBR dome. Okay. This is a very special thing which is having a heating. This is also having a heating here. This base is a metal base, and this metal base is for heating purpose. Now there is a one inlet for water, outlet for water. There is a one injection port is also available. Now in this case, this is a pyridine attachment. You can say the pyridine injection port. Now this I am showing you. This is the thing which is used for. This is the thing which is used for the pyridine injection. You can say that the pyridine injection. So you can inject the pyridine from here. So if you see, it is having like the 4 hands, 1 nose, water inlet, outlet. So we used to call it as our Ganpati idol actually. See, this looks like an Ganpati idol. It's a very simple thing that is there. Okay, so this is actually the instrument. Now here, if you see, there is a one line, gas line is also coming. So it requires a nitrogen. So it requires a nitrogen as a carrier gas to carry the pyridine to the sample. And if you see, the sample is here. See, inside that dome, there is a sample. this dome is made up of KBR, and this KBR is too costly, and that's why this is very important to understand. Now this is the material which you have to be analyzed, and this is kept inside this dome. And now you are injecting the pyridine. This pyridine flows with this nitrogen gas, and then it gets in contact with gets in contact with this material. And if that material is having Bronsted acidity or the Lewis acidity, according to that, it gets attached to the material. So pyridine gets attached. And remember, this is a completely qualitative analysis. It is not the quantitative. And we want qualitative only. Remember, the requirement is only the qualitative. And distinctly, you can identify which is Lewis, which is Bronsted. So this is actually the instrumentation part. You can get it on the net also if you go for the Bruker instrumentation, but they will not show you the details how the sample is there. They will ask you to come for the training and so on. So this is actually the praying mantis which is there. Now understand this logic. Understand that why we are putting the pyridine. So we are putting the pyridine because pyridine is a base, and it will take the proton to get the pyridinium ion. Understand? So if your material is a Bronsted acid, remember, if your material is a Bronsted acid, it will take the proton to get a pyridinium ion. Pyridinium ion. Remember. Now this is a NH stretching. Now this stretching is important for us. Now this NH bond, this NH stretching is important to us. And that's why this pyridine studies is important. If your material is not Bronsted, you will never get the NH stretching frequency. And you can come to the conclusion directly that my material is not having the Bronsted active sites. Similarly, you have the pyridine is a Lewis base, and when it is coming with the metal, in contact with the metal like ruthenium, iron, indium, iridium, or you can say aluminium, it gets coordinately bonded. Coordinately, remember again I am saying coordinately bonded, and forms N metal bond. Nitrogen metal bond actually, and this is a Lewis acidity. Remember, this nitrogen metal bond comes in the specific region. Where it comes, that we are going to see in the. But the thing is that we can have whatever the material you have. That is the support. On that support, we have the active species like Bronsted acid or Lewis acid, and is whether that Bronsted acid is there actually on the support so that it will act as a catalyst. That has to be confirmed. And if you want to confirm it, you have to do this type of characterization. So the first question will be coming into the paper reviewers comment that you have prepared the catalyst, you have done the dispersion of Bronsted acid or the Lewis acid, how you have confirmed it. And if you have confirmed it, just give this evidence or the support for this. So in that case, then you have to give the pyridine IR is the one support which is very mandatory, which is very essential for the satisfying the query of the reviewer or the editor of the journal actually. Okay, so this is the moderate thing actually. Okay, so now these are the first study of this pyridine IR was carried out by the E.P. Parry, and it was published in the Journal of Catalyst in 1963 actually. And there comes now a lot of version of these studies. Of course, so many people have done so many innovations and novel things have been done actually. But there are now so many studies which are coming. But basic study is, suppose there are three types of possibilities. Am I audible to everybody? Am I audible? Just am I audible?
Yes, Sakti sir, yes.
Yeah, okay, sir. Okay, thank you. So this is pyridinium ion formation is one place. Coordinately bonded pyridine is the another place, another possibility, and hydrogen bonded pyridine. So if that pyridine is not actually having the hydrogen attachment, no. But very specifically, I am saying, if your material is a Bronsted acid, I will just explain with the very simple understanding. If your material is a Bronsted acid, it will show you a very strong peak at 1540 to 1545 centimeter inverse. 1540 to 1545 inverse. If your material is a Lewis acid, you will get the peaks at 1440, 1490, 1590 and 1600. Lewis acid, 1490, 1590. Pyridinium ion, 1540 is a very characteristic peak of pyridine iron, and all these are the secondary peaks which are observed along with the material. But pyridinium ion, very specifically pyridinium ion will come along 1540 to 1545 centimeter inverse. Okay, I am actually at 2:30 right now, but as we have started 15 minutes late, I will be continuing more for 15 to 20 minutes with the organizer's permission. Okay. Okay. Now see, this is the first thing which I would like to put in front of you actually. This is the Bronsted acid HPA supported on MCM. See, which we have, what we have done, we have taken a MCM 41 and we have supported the with it on HPA. These are the heteropoly acids which are there. And if you see here, MCM 41, only if you take a support, only if you, these are the origin plots of the IR data which we have observed. So MCM 41 does not show any absorption band at 1540, because it is not Bronsted. Now, if somebody will ask me, sir, it is not Bronsted there, that means what? It is a Lewis acid because it is a silica. MCM 41 is only silica, and that's why it is showing only the Lewis acid bands or Lewis acid bands at 1440 or 1440 and 1590. But as your loading is there, 20% PWA is loaded here. You observe here that you are getting a band, getting sharp band at 1540. 1540 band is observed here. Plus 1490 band is also observed. Now, why these bands are observed? Because PWA, original PWA shows the spectra like this. Original PWA original phosphotungustic acid. Original phosphotungustic acid is itself the Bronsted acid. So that's why it has to show the Bronsted acidity in the catalyst, and we can confirm that we have prepared the catalyst because PWA, to use only PWA is not possible because its surface area is very high. Its activity will be very less. Its TOF and TON number will be very less. But if you have a support, you can have a very good dispersion of PWA on the support of MCM 41, and then you can have the very good activity for your catalyst. And that activity you have confirmed by using the pyridine IR studies that my compound is showing the Bronsted acidity, which is observing the peak at 1540. This is actually the work which we have published in the New Journal of Chemistry, actually. And this is actually the organic reactions which we have performed. So I have just reported 15, but for your information, I can tell you I have done the 60 or 50 samples deprotection study. This is a deprotection of organic compound. So now ethers are converted into phenol. Simple organic chemistry is there. So these are and selective simple organic, selective. In the presence of other functional groups or other ether groups like OCH3, only we are getting the deprotection of allele. That is the very beautiful part of this which we have studied, and that was reported in the New Journal of Chemistry, Royal Chemistry Journal, Royal Society of Chemistry. This is the journal. This is again another one, which is the Lewis acid form. Now see, this is again MCM. This is MCM is a support. This is the support MCM41. Now this MCM41 support we have loaded with FeCl3, iron 3 chloride, anhydrous iron chloride, anhydrous FeCl3. Now this anhydrous FeCl3 we have put on it. Now if you see, it is having the Lewis acidity, but iron chloride, somebody will say, sir, it is a Lewis acidity, so it is showing only 1480, 1490, but it is showing the Bronsted acidity also. 1542 centimeter inverse. Now somebody will say, sir, you have shown Lewis acidity and that peak is only for 1440 to 1490 centimeter inverse, and now you are saying that this Lewis acid supported on MCM41 is showing the Bronsted acidity also. This was a very accidental thing which we have observed that in our studies while we are discussing with the Rode sir, that it is found that whenever you are treating with the Lewis acid with MCM41, there is a generation or the formation of HCl also which is observed, and that HCl which is observed is used, which is there, and it was observed as a Bronsted acidity. So this catalyst we have prepared from the point of view of Lewis acid catalyst, but it is actually the Lewis acid as well as the Bronsted acid catalyst, and HCl is generated. Of course, the formation of HCl is very less, but it is also generated there, and which is useful for the organic reaction. Now what I have done a very simple organic chemistry, that a Claisen rearrangement we have performed, because the Claisen rearrangement requires the Lewis acid. That role was played by the FeCl3, and it requires the H plus for the protonation. That role was played by the H plus, which is formed during the formation of the catalyst. Now see, we have done about 10 compounds, different organic compounds, which we have prepared as a dihydrobenzofuran synthesis. We have not just done only the Lewis acid, it is the Claisen followed by cyclization. So we have done a dihydrobenzofuran synthesis actually. So this is the another very important class of the catalyst, that we have prepared the Lewis acid supported catalyst, we have prepared the Bronsted acid supported catalyst. Then I have worked with the Pawar sir also. One of the student, Neeraj Kumar Zadav, was working with us, and he has prepared the doped material. So it is not only the Bronsted acid, Lewis acid, this is the doped material actually. And if you see that, the very important part of this point is that as the doping goes on increasing, the Lewis acidity of the catalyst goes on increasing. See the doping, this is only the pure ZNO. This is the ZNO which is having the titanium doping. This titanium doping is increased. Now see, you will see that this is increased. This area is increased, and this is only normal. Using this catalyst, we have done a variety of compound synthesis for Friedel-Crafts acylation reaction. That is the dyes. This was the fantastic paper which was published in the catalysis communication actually. So we have done the simple chemistry, that is alkylation chemistry, but we have used this type of techniques to understand the acidity of the compound actually. This is also another same catalyst, but this is published in the ACS Omega. But because we have we have in a position to prepare the catalyst which is having the Lewis acidity, which was confirmed, and this technique was used for this type of thing. This is the PWA, that is polyvinyl pyrrolidone actually. So this is a phosphotungsten. This is a one sort of hybrid material which we have prepared. That is the that you can say this is simple. Just a minute. This is simple material which we have prepared pyrrolidone, and this is also used for the alkylation reaction. So this was confirmed. Again, if you see, this is having a PWA, normal PWA graph. These all are origin plots. Then this is about the PVP PWA. This shows the very good Bronsted acidic character in 1540 centimeter inverse. So you can have the simple ATR with you. That also, if you have ATR, that can be also used for the pyridine IR actually. But you need the very early pre-treatment for doing the pyridine IR studies on ATR. But the normal procedure is that you should use the pyridine IR technique by using this type of praying mantis attachment. And this praying mantis attachment, nowadays, very few pyridine IR are also available and working. Perkin-Elmer service is very bad as per my knowledge, and that's why this Perkin-Elmer is not good actually for the service point of view. But now Brooker is also giving the praying mantis attachment. So now this point with this point, I would like to come to the conclusion that you can design the different catalyst of Bronsted acidity, Lewis acidity, and you can use this catalyst in the various organic transformations at your college level also, and you can design the different types of catalysts. Thank you very much for giving me this opportunity to speak in front of you. And now I would like to answer few questions, because I'm expecting that, because this technique maybe people are knowing, but I want to know that whether there is any doubts. Yeah.
Okay, dear delegates, please ask some queries or doubts regarding this technique to the successor, and I hope successor resolve your query.
Yeah.
Unmute your mic and then ask the questions. Okay.
राठोड सरांचा माईक बहुतेक ओपन होत नाहीये. राठोड सर.
यस सर.
हा येस सर. येस सर. येस.
तिसरा.
हा येतोय आवाज येतोय.
बोला.
आवाज येत नाही सर. आवाज
हे होतोय त्यांचा.
या त्यांचा आवाज येत नाही.
ओके. अदर आय रिक्वेस्ट अदर पार्टिसिपंट कम फॉरवर्ड. Okay, there is a nobody.
नाही, त्या राठोड सरांना काहीतरी प्रश्न विचारायचा आहे.
त्यांचा आवाज होतोय,
पण त्यांचा आवाज येत नाहीये त्यांचा.
बघूया. ठीक आहे. Okay, dear colleagues, dear participants, now Dr. Sachin Sakte from the Modern College delivered the lecture or talk on the IR pyridine. Now it is especially the new for us. We working on the heterogeneous catalysis at that time. The characteristics of catalysis is a very difficult problem for us. Therefore, Dr. Sachin Sakte give the cooperation regarding the carry out the characteristics of our catalyst by using this pyridine IR, ammonia TPD, and nitrogen absorption. Therefore, these characteristics are generally carried out by this successor, and that's why our papers are published in a very good journal. That is CATCOM. Second one is the ACS Omega. Third one is the TETLET, Tetrahedron letter. Therefore, there is a linkage or collaboration established between our group and Sachin Sakte's research group, and there is a beautiful, or that is a good publications are published in this reputed journals by the renowned publisher. Navy Springer is Omega. They also published in this ACS and also in elsewhere. In future, there is a collaboration is continued. If any participant want to need to carry out the characterizations of the catalyst, we also take the help of the successor. He is always ready to cooperate the researcher and also guide the any type of this difficulty arising.
I want to add here. We have incorporated this technique because as an autonomous college now, we have designed a separate course for the analytical chemistry peoples where solid UV, solid IR, solid NMR, XPS, XRD, low angle, wide angle. A separate paper for the characterization of catalyst is designed, and that freedom we got only because of autonomy. So that's what again I want to say, that you should also design or you should also plan to put this technique so that the student, when he will go for the research, he will come to know that these are also the techniques which are to be studied for the further PhD degree or the MPhil degree if he want to pursue.
Sachin, definitely we take your help, and next year we are going to autonomy. In that next year, we also try to develop one course in collaboration with you for our analytical student.
Sure,
and it
sir.
also benefited for our researcher. Our college also has a research center.
Sure, sir. Sure,
therefore, it is a collaborative work, go on a hand in hand. Then it will achieve also benefit to our.
Rathod sir is still want to ask, but he is mic or something.
Sir, yes, I have some problem, but I will personally meet you in Pune, sir. Whenever I meet here.
No, yes, sir. No problem, sir. No problem. Yes, sir. Great, sir. Great. You are always welcome. Always welcome. Yeah.
Thank you, sir. Thank you.
Okay, Santosh, if your mic is on, please can you do the bottom check?
Yes, sir. Already on, sir. Mic is on. Yes, sir.
Okay, good.
Okay, sir. Sir, I am grateful to the organizer for this opportunity to me. In this session, we had an opportunity to hear the lecture of young and dynamic resource person, the Dr. Sachin Sakte, sir. Sir, today you have shared your valuable knowledge and experience regarding this new technique, that is pyridine IR instrument, with us. This will surely be going to encourage us, sir. This is new technique for us. On the behalf of all participants, the organizer and team associated with this FDP, I am very much thankful to Dr. Sachin Sakte, sir, for delivering such a nice and informative lecture for us. Thank you, sir. I am also thankful to the organizing team of Professor Ramkrishna More College, Apodi, UGC HRDC, Savitribai Phule, Pune University, Pune. Lastly, I am also thankful to all the participants. Thank you, sir.
Thank you.
Okay. Thank you, Santosh, giving this vote of thanks on behalf of our college and HRDC. And again, I also thank you.
Thank you.
Sachin,
thank you, Nagne sir. Thank you, Nagne sir. Thank you.
Okay. Thank you for coming and spare the time with us, and also discuss the pyridine IR technique for this useful for our coming budding researchers in a biological and chemical sciences. Okay. Now we move to next session. This is again last session is a technical session. Now a resource person from the Savitribai Phule Pune University Central Instrumentation Facility the Center of Savitribai Phule University. There is a Pradeep Kharade. Kharade, sir?
Hello, am I audible?
Yes.
Okay, great.
Welcome for this session. I'm Dr. Arya Pawar from Ramkrishna More College, Akurdi. Welcome to you for this session, and I request to you the whatever information regarding the CIF, please deliver to our participants.
Okay, good afternoon, everybody. Am I audible?
Yes, audible, sir.
Okay, okay. So,
Yes.
okay, can you see my screen?
Yes, sir. Correct, sir. Screen is visible.
Okay, thank you. So it's a big challenge after lunch to deliver something, regarding NMR instrumentation. Well, this is a bit off session, I would say, which is related with the instruments, but not really the earlier lecture has been covered. This is something researcher should know actually, what we can do with the instruments, right? So this is the attempt for that. So before starting to this session, actually this came to us in a very short notice. Yesterday evening only we have received this notification, but we will try to make it very comprehensive and useful as we can. I do have my team with me of the CIF and chemistry department. So let us start. So as you know, in the NMR there are the three basic parts. One is the magnet, second is the probe, and third important is the data control and processing unit. Serially, what happens over the years, each and every part of this particular instrument has been changed a lot because of the progress in technology, and the earlier instrument and the instrument today are totally different. So let us have a look how it was earlier. I would like to show you some very interesting permanent magnet instrument here, and this is by Perkin-Elmer. Maybe many people may not be aware that once upon a time Perkin-Elmer was manufacturing this kind of NMR machines. And you can see the line, this line is important, that this is actually an advertisement. Perkin-Elmer permanent magnet NMR spectrometer operate for only pennies a day. So this also has been changed a lot over the year. It was working for pennies a day, and whereas today's instrument require a consumable of thousands of rupees per day. So that is the biggest change. Then I would like to share this particular model with you, model R32. This is the permanent magnet NMR instruments which we had in the early 90s or late 80s, I would say. So this was the instrument was working till that time, and it was a continuous wave permanent magnet instrument, and lot of researchers now spread worldwide in the very high positions. They have used this instrument, and Professor Dawde sir was in charge of this, and he really operate and made lot of things on this particular instrument. So this is something I would like to share with you. And this is, Department of Chemistry is fortunate enough to have all the three versions of the instrument, and so am I, that I could maintain all these three versions of instrument, three generations of instrument, where it was a permanent magnet. And this is what permanent magnet instrumentation was, comparatively very simple, but at that time also it was very challenging. You can see these two magnet poles over here, Then, as I said, this was a continuous wave instrument. So there you required some sweep coils over here. So these are the sweep coils. And then you can see, of course, there is a sweep generator. And you can see here the radio frequency transmitter and receiver amplifier in two different formats. So one is the transmitter and one is the receiver. And ultimately it amplifies, then it goes to the processing unit, and you will see the signal on oscilloscope, and you will record the signal on recorder. Okay, XT recorder was there, and on that recorder you can record the signal. You won't be able to save the signal or save the FID. That kind of facilities were not available at that time. So once you get the hard copy, that's all. And then there was one popular post known as draftsman, and they used to, trace all those things, and you can publish into the papers. Then this was the second instrument we had, FX90Q. Image is not clear enough, but you can see here it is a magnet, and it was a electromagnet. So the difference between the earlier instrument and this instrument was mainly this magnet. So electromagnet, you have to give the huge power to this electromagnet, and because of that it used to get heated, and for that compensation you required a chiller outside, which will which will dissipate the heat generated into the magnet, and will compensate that particular heat. And this is how the magnet is generated. And then once the power is off, you can switch off, the magnet gets switched off. Then another point over here was that this was a light pen arrangement was there, and a bit advanced data processing unit was here. So this is again then this was FT instrument. And as you know, there was a revolution in the magnet technology, and then superconducting magnets were introduced. So permanent magnet and electromagnet has got their own limitation, and which can be run up to the strength of 2.35 Tesla, with the help of which you can build an instrument not more than 100 megahertz field. So this is how the strength has been increased, and in superconducting magnet nowadays you can get the strength 21.15 Tesla, and you can manufacture the instrument 90 megahertz or even more. 1.2 megahertz instruments are also in the pipeline. So this is how the technology changed for the magnets, and simultaneously for data processing and other. Let us now a very interesting video by Brooker I would like to share with you. This clears all what exactly how NMR works, and how it really gives you the signal. So let us see that. I hope it should work. Yes, so here you can see this is from the Brooker. We do have a Brooker machine, and very nicely it has been explained. So this is a superconducting magnet, this is the console, and this is really the inside of the magnet. So as you can see, liquid nitrogen chamber is here, which is the outermost one, and inside that there is a liquid helium chamber, and then superconducting coils are placed over here, and which are immersed in the liquid helium. So you have to keep the magnet alive with the help of liquid nitrogen every week and liquid helium once in 6 months, depending on the design of the magnet. So this is how the magnet look. I mean, this is the you can say inside view of the magnet. And let us move ahead. And of course, all these chambers are divided with the very high vacuum, which will control the evaporation of liquid nitrogen and liquid helium. So let us see next part. So from here, sample goes in and it really sits in the coil. So these are the coils. So as you know, this is required to be placed where you will get the highest strength of field, and very important is the homogeneity of the magnetic field, which is an extremely important component when you record the NMR spectra. Homogeneity of the magnet is very important. So these are the radio frequency coils. As in earlier slide, we have observed that in earlier instrument, there used to be 2 different coils for transmission and receiver. So in new NMR instruments or in the latest NMR instrument, only one single coil which is used for trans receiver. So same coil is used for the transmission of the RF pulse, and the same coil is pick up the signal from the sample and it will give you the spectra. So this is the major difference between these two. So here you can see 3 types of signals are produced, and the difference between these 3 signal are like the difference in the frequency of these 3 signal is different, and also the amplitude of these 3 signals are different, and this is then calculated by the FT, and then effectively you get the NMR spectra like this. So this is how the frequency domain is converted into the time domain, and you get the spectra, and this is quickly it shows that how multiplets are. So this is how you get the NMR signal recorded. And as you can see, this is a typical NMR room in which you can see the superconducting magnet, then data console unit, then this is the operator's chairs, and this is where you get a data process door here. And these are the typical signs, like when you go to the near to the superconducting magnet, you should not have any magnetic substances with you, like people with pacemaker may have a problem, and so on. Now there is also one big change has come up. This was the earlier scenario, very strict, but nowadays these superconducting magnets are coming with the shielding. So the effective magnetic field reduces, outer field is reduced by giving the counter magnetic field to the superconducting magnet which is there in the instrument. So this fear has been little bit reduced, not exactly fully, but little bit reduced, and now it is more safe, I would say. So this is how the typical NMR instrumentation setup is. As you know, this is all the instruments are network connected. So this is nowadays a very common feature to all the instruments. Just now we have seen what is there inside the magnet, and this is how the probe looks like seated over here. And as far as instrumentation part is concerned, this is the acquisition and processing, which controls the magnet and signals also. So from here it transmits the pulses to the probe, and it also receives the same the signal from the same coil and sends to the acquisition and control unit. This is something different. BSMS, the BSMS, this is because of the Bruker, so it is Bruker shimming and management system. So this is how NMR sample looks like. We are going to have a look how NMR sample is required to be prepared and all. But let us see, this is the sample tube. These are the spinners, and this is called as a depth gauge. So this depth gauge is calibrated with the help of, with the instrument, so that this shows you where you will get the best and stable and strong magnetic field. This guide really gives you the idea. And spinner, this sample tube is properly placed in the magnetic field. Here there are some adjustment screws. Over the period you can check the field density and stability. Accordingly you can adjust those. So this is how the sample tube and spinner combination looks like. And this is, as I said, this is the guide by which you can fix the sample height. So there are different types of consoles nowadays available. We do have 300 megahertz instrument and 400 megahertz instruments. So this is the instrument Avance 3D we do have, with the 300 megahertz superconducting magnet having 2 channels, and this is the one for the 500 megahertz instrument. This is 400 megahertz instruments. 300 already we are going to running down. So this is 400 megahertz instrument, and this is 500 megahertz instrument having the different channels. And these are the advanced console looks like, where you can have a 4+ channels. This is called as a double bay systems. Now let us see a bit of electronics, how it looks like. So this is the red is the transmitter path, and the blue is the receiver path. So this is the duration of the pulse is important, which is called as a delta t, which is determined over here. Frequency, then phase of the signal, the waveform is required to be controlled, and of course the amplitude is required to be controlled, where it should not be very high and too low, it should be optimized. And this is how RF pulse is generated, which is given to the probe, as I said, a single coil. And then this part is very important, which is called as a duplexer. So once the RF pulse is fed to the probe, after some time delay, it is switched by the duplexer, and then it starts collecting the signal. So while collecting the signal gets amplified. Then there are the filters, so unwanted frequencies are dropped out, and then of course A to D converter for the Fourier and other transformation, and then there are digital filters, and you get a very nice NMR data through this console. Okay, so these are the systems looks like this. This is the typical probe probe structure. This is the real photograph of the modules of the NMR system. There are different modules here, as I said, signal generation unit, where frequencies, pulses are created as per requirements. This is the receiver path, again same from the probe. There is one unit situated very near to the magnet, HPPR, and with the help, because the signal coming, the receiver signal is very low, and it is required to be travel as minimum as possible. So there is one unit over here where you can see where you can collect the signal and get it amplified, filtered, and fed to the RxD next for the further processing. Then let us have a look, inside again. This is again the magnet. These are the magnet coils immersed in liquid helium. The temperature is 4.2 K. Okay, so it is around -269 degrees centigrade. So this is how it looks like. These are the cryo shims. The concept of cryo shims is that these shims are immersed in the cryogenics like this, that is in the liquid helium, and you get a best homogeneous feed with the help of cryo shims. When you have good number of cryo shims, you can get a very stable magnetic feed, stable feed. And these are the shim coils, outside room temperature shim coil. So as I said, the shimming is very, very important part in case of NMR. You can see over here, this is the shimming system, shims, how the shimming is important. These are the different, you can say Z1, Z2, Z3 are the numbers given to the shimming coils. And when you adjust these coils, you will get the stable magnetic, homogeneous, stable magnetic field. So what really means the bad shim and good shim? With the help of good shimming, you get a very good sharp signal over here, and the line width also comes down, and you will have a good amplitude of the signal. And in the FID, actually, this is the decay rate, and you can see this is in the FID. You should cover as much as possible the signal area so that you get a good signal. So this is achieved with the help of good best shimming. There are n number of probes available nowadays. as per the sensitivities and as per the requirements. The most common used probes are room temperature probes, which can be used for the solid state samples also. There are special probes for the solid state. Even there are HRMS probes, so you can, use the MS in coupled with the NMR. Then there are flow probes. Other than normal probes, you do have a flow probes. So continuously you can flow the sample through the NMR system, and you can scan the spectra. These probes are very expensive. I mean, it costs the as good as one new instrument. So these are the very expensive probes, I would say. Helium cryoprobe and all. The maintenance cost of this probe is very, very high. So exactly what happens when tuning and matching of the signal is very, very important. And what exactly this tuning and matching? You can see there are multiple nuclei over here. Okay, so this is say proton, this is C13, this is phosphorus, and so on. This is fluorine. So this is exactly like, your old wall type radio sets. If you want to listen the Pune Akashvani, then you have to tune your radio set to Pune. So same is the case. If you want to look at the proton molecule, then you have to tune your probe to proton. So as you can see, this is the frequency 400 megahertz for the proton over here. And then if you want to tune it to C13, then of course you have to change the station. You want to listen the Mumbai, so it will look to the C13. So this is how the tuning and matching of the probe. And if anyone has used this kind of radio, they might be aware how it was difficult to tune your set to particular station. It was something, a job of some skill. Same thing applies over here. So, but once it is tuned, then you can go ahead. So this is all because of there are inductive and capacitance circuits incorporated in the NMR instrument. So then, as far as regular maintenance is considered, you have to fill the liquid nitrogen. So as you can see, this is magnet information system, and you can see here the level of liquid nitrogen. You can see the plot over the days. These are the weekly refills. This is the liquid nitrogen cylinder. Then one output goes to that. You have to pressurize your filling cylinder, and flow of nitrogen will start. You have to pressurize the nitrogen cylinder. You have to take a lot of care, minus 180 degrees centigrade. This was the older technique when you have to dip the, dip stick, and you will come to know what is the liquid nitrogen level in the instrument. But now these days are gone. Digitally, you can see what is the level of the liquid nitrogen. So this is one that liquid nitrogen. Again, the next part is the liquid helium refill, which as I said, minus 269 degrees centigrade. Current cost of the liquid helium is rupees 1,500 rupees per liter. So at least you have to pump 1.5 lakhs to 2 lakhs for one refill. Again, this is the older technique to measure the liquid helium. nowadays you can really have a digital output. This is how the helium plot looks like. This is very crucial part, and you have to monitor the liquid helium always, helium level always. Otherwise there is a fear of quenching of the magnet, and it will cost you another 15 to 25 lakhs of rupees. So this fluttering shows that you have reached to some stage of the liquid helium refill. This is again, as I said, this is an older technique. You can check the upper and lower level of the helium, and from that you can, I would say, have a guess of liquid helium, physical measurement of the liquid helium. This is how the actual helium refilling goes. You have to remove the helium port and keep it open. Then you have to immerse the liquid helium refill tube in the liquid helium container from which you are going to refill it. So as it goes down, these are the two levels which we observed last time in the last slide. Again, you have to pressurize the liquid helium and see how this liquid helium is coming out. Minus 269 degrees. This is called as a pre-cooling of the helium. Pre-cooling of the helium line. Now carefully one has to insert it into the magnet, and this is a job of expert only. Last 20 years we are maintaining this kind of magnets on our own. There are very simple things you have to always monitor while liquid helium refill. Otherwise there is a fear of quenching of the magnet, which that accident would be very, very costly in terms of human or even for the instrument. So this shows that your magnet is full, and now you should stop immediately to refilling the, otherwise whole helium will come out within few minutes. So you have to stop at a particular time when it gets refilled fully. Pressure has gone down. No flow. That means, it starts whistling, means your container is empty. So as I said, the next video is about the magnet quench, which is also very more than 100 ampere current is always present in the magnet, and because of that, if there is any thermal instability in the magnet, then it will start heating and it will go in the loop. So one small point will hit all other points, and slowly the evaporation of the liquid helium will start, and at one point the magnet will get quenched like this. So if this kind of quench, this is called as a soft quench, then it's also okay, but sometimes the magnet may get explode. So you can see this is the soft quenching of the magnet. All the 3 ports are open, and oxygen level in that particular room will go down. So this is how this is about a quick look over the NMR instrumentation. We do have a 400 megahertz instrument and 500 megahertz instrument. These are the charges. You can visit to our central instrumentation facility. And apart from NMR, we do have other instruments like ICP-MS, PPMS, then LC-MSMS, GC-MSMS. We do have a FEACM, which is also being used by many researchers. We do have single crystal XRD, confocal microscope, and so on. You can visit our site cif@unipune.ac.in. You can give the Google search and you can reach to us. Now let us go to the real NMR room, and Sheetal is there, I believe, and she will show you how the sample preparation is made, and what are the facilities available really on the NMR instrument. So over to Sheetal for 400 megahertz Bruker instrument. Hello, Milind. Can you take over?
Hello. Please allow Sheetal madam to speak.
Hello.
इथून नहीं करूँगा.
Hello.
बंद करून चालू करता का इधर.
अनम्यूट नाही होते. अनम्यूटच नाही होते. आवाजच येत नाही.
नाही, एक मिनिट. याने सुरू करा ना. याने सुरू करा.
याच्याने का?
सॅम्पल प्रिपरेशन दाखवा. Hello. Are we audible now? Hello. Now Sheetal will show us the sample preparation. Yeah, okay. Please go ahead.
Hello, good afternoon, everybody. Now I'm going to how to prepare sample for animal analysis. We have one sample, one standard sample for nitrogen alcohol, which is in weight is 5 to 10 mg. For H1 we require 5 to 10 mg sample, and I already take 8 samples. And for animal we require different solvents. Now I'm going to prepare the sample. This is chloroform 3N3. This is DMSO, dimethyl sulfoxide. This is D2O, deuterium. Acetone and methanol also we can use here. So for the H1, we require 5 to 10 mg sample, and for C30 we require at least 40 to 50 mg samples, and it will be completely dissolved in 0.5 ml of deuterated form. So animal will require saturated form because we use normal solvent. Then protonocular solvent. Sample proton. So you cannot analyze, you cannot get proper spectra, means intensity of the solvent peak. It will be very high. So you cannot interpret your sample compound. Approximately about 0.5 ml. Is completely soluble. Basic requirement for analysis is like it is completely soluble in proper solvent, and should not be there should not be any particles. If there is some particles, we will slightly vomit by this machine, and even though that again is not visible, that time we filter it with this help of this funnel by putting the paper on this, and we transfer this sample with this. Then we get the clear solution for NMR analysis. So there is no need of this funnel right now because this is a completely soluble sample. So I'm going to transfer into the NMR tube. Before that, we have to just check whether this NMR tube is completely dry or completely clean. Should be clean and dry. This NMR tube is having a diameter 5 mm. Is being called NMR tube like this, you can see. And the best precaution is this: it should be completely dry and completely clean. I'm going to transfer this into the NMR tube. Only 5 NMR solvent. This much concentration is only we need for H1. If we take more samples, high concentration samples, that time we not get this spectra because resolution the spectra is low and get broad spectra will not get proper splitting. I will show you how this is coming if the resolution will not be. Sample this I'm going to put sample this then clean the NMR tube by outer side of the tube. Then this NMR here. This is the spinner, also called as folder. You have to just like this, and NMR tube should be like this. And you have to take care of that. So NMR tube should touch. It should be touched to this part of this folder, and this both folder touch to each other. It should not be like this. It should not be like this.
Yeah, so present code.
Now giving the command.
The presentation.
This link first.
Digital Marathi. Okay. Am I audible?
Yes, sir.
And can you NMR documents? I think you. I want to present process, and once acquisition is start, you can see the acquisition mark over there. So I don't know why it is taking such a long time. Okay. So point is that to show you the how the NMR software look likes and what really operator does over there, and once you give the sample, what precaution and what are the prerequisites for the sample are there. So this is what the point is, and the different type of experiments we will again show you. I don't know why it is taking a bit long. It takes time for streaming. Yes, so you can see the streaming has been achieved. Show in top screen, you can see so acquisition is in process. These are the status of the lock. Okay. So here you can see the locking is good.
Scan out of 16 scan, how much scan is completed here? It is one will be completed. After one scan, we can see the experiments spectra also. This is the processing commands. Transfer FID to
Thanks, Subramanian. How it looks like.
So this is the raw spectra, and she will phase it now. And this is how you will get the NMR spectra on your screen.
See.
So this is how the doublet looks like. Now, actually, I would like to show you the number of the experiments which we can do on this instrument. So let us switch to that window, Sheetal. Yeah, I mean, automation window. Okay, and if you, this is how, you can see the list of experiments. There is a big list of experiments by which you can, this is the basic proton H1 experiment. It starts from that, and then, okay, acquisition has been completed. Let us see the final spectra. So, can you show the full spectra? Okay. So, this is how the spectra looks like. All the acquisitions have been completed, and you can see the information window is now not updated, so everything has been done. Now let us go back to the list of experiments. So if you click, as I said, this is the proton, and after proton you can see n number of things, which is QC, code Z, no Z. Probably tomorrow you are going to have a lecture from Professor Dave, sir. So at that time, he may explain or give the. So it's a big list of experiments which you can do on this particular instrument. So this is, and nowadays, this instrument is online. Online you can see all the things. So probably one can operate the instrument also online. So that is what the possibility nowadays. Of course, as a special case, we have connected this instrument to the internet. Otherwise, we won't, because if there is a virus attack or something goes wrong with the software, it will cost us not less than 5 lakhs rupees. Okay, so this is how the this software and all the things look like. Anything you want to add, Sheetal? Sheetal will add something, some quick.
More experiments I will show you. Just now, yesterday. We have these 2D experiments. Yesterday I done it. Cozy experiments, you can see here. On the cozy experiments, we can see H1 on both the side. By correlating this peak, we get the directly H1 bonding. Now we have HSQC experiments also. Means in that H1 C13 correlation we get like this. So directly you come to know that which hydrogen is connected to which carbon. Then we have also temperature dependent study. So you can see here. See, like this, we have temperature dependent study. At different temperature, we took this NMR, and how changes happens in this NMR. You can see this shift occurs like this. Other peaks are not shifted, but this hump is shifted to the off field. This is the effect of temperature study. Then we have also titration study also done here. You can see the titration. Titration. So from this we can get more information about our sample. We can get more and more proper structure, find out if this is experiment. Titrations, reaction. Yeah. This is titration study. So, in this also, see the change of this peak. The peaks are 100 position, but one is multiple peak. It's shifted here. This is titration study. From this information about the sample and you get the chemical structure more and more proper.
So the point is that we just want you to introduce what things actually you can do with the NMR instrument. So let us have a separate and we will have one more quick visit to CI. So thank you very much, Sheetal. Okay, can you hear me now, all others?
Yes, sir.
Okay, now I'm going to show you the, this is how our NMR instrument look like. This is, as I said, the 400 megahertz NMR instrument, the software of which you have just seen, just installed in the Department of Chemistry. Very tiny console. And here we do have an old 300 megahertz instrument. The superconducting magnet is still live. However, we are not using this instrument nowadays because this is the most advanced one we have received. This instrument we have to run down because of the expensive cost of consumables, liquid nitrogen, liquid helium. As you can see, these 3 liquid nitrogen cans are there. So you should have always one in the stock. This was really a good instrument for Department of Chemistry and lot of researchers worked on this. So let us now visit to Central Instrumentation Facility. So all of you are invited to visit the Central Instrumentation Facility, but this is, I would say, a virtual tour. Something is going on for the safety. You have to have fire extinguishers, so recently being replaced with, and you are entered here in a very good setup. As you can see, we are entering into the NMR room, 500 megahertz FT-NMR. So here we do have a good team of operators, and this is really the workhorse for our Central Instrumentation Facility. Here is the Neha just now preparing the sample. Then Snehal has very long experience in the NMR operation from the NCL also. She has been worked in the NCL, and again newly joined Dheeraj over here. So let us see this 500 megahertz instrument NMR. The major difference, this instrument is with the facility of auto sampler, which is called as a sample express, as you can see. So, Snehal, can you load some sample so that we can see how really the auto sampler works? So this is the auto sampler. You can load samples, and this will work for you, over the day and night. So 60 samples you can load. A sample is being selected, and then sample is being inserted. It has been inserted. It goes down. Probably this is the advantage of virtual tour. Even if you visit here in the central instrumentation facility, you won't be able to see this kind of automation because this is very top. You can see. Okay, fine. Can you change the sample once again so that this is very interesting. It will take some time. Okay, fine. That's good enough. But okay, let us see. Some part of the software we have already seen, and this instrument, of course, we do not connect to internet, so definitely we can't take risk of showing you the software online. But let us see. So this is the same software you have seen just before on the 400 megahertz instrument. And Snehal, can you show us some spectra? Probably shared spectra, yes. Now, with the help of this instrument, we are catering to many industries, and many researchers from the different colleges are using this instrument. So this is the proton spectra. Then this is the laser printer. These are our updates. What is the liquid nitrogen level? So Dheeraj, Neha and Snehal are updating this information on the board also. So this is how the total setup looks like. Neha has opened the console door. Thank you, Neha. And you can see the console of 500 megahertz instrument. Okay. So this is how it looks like. As I explained you, this is the SGU, which I showed you, signal generation unit. It creates the pulses, and this is the HPPR, as I mentioned. So here you get the transmitted signal, and that has been amplified. So this is how the console looks like. This is the data processing unit modules. This is the main power supply, and this is how the instrument is. Let us have a quick tour of LCMS also. We do have Nitin Kadam with us, and he will quickly introduce you about LCMS instrument.
So this is LCMS instrument. This is HPLC, and this is ultra high pressure liquid chromatography. So in that you have seen the HPLC in your laboratory. So there will be a solid reservoir. This is our pump through which this is one pump. This is second pump. So through which mobile phase is passed, and this third compartment is auto sampler. And after that we can join here in the column. Okay. So what is column? This column is C18 column. Okay. This is a C18 column, and particle size of the column is 5 micron, and whatever the pore size of the particle is 120 angstrom, and what are the dimensions of the column? The length of column is 250 mm, and internal diameter of the column is 4.6. So we attach this column to the inlet, and there is an arrow mentioned. So like this, so we can attach column, and we can have if you have your compound or impurity profile, there might be some impurity. So that can be separated by this, and this is
This is a Q-TOF, time of flight. So that's why the length of this detector is very high. So some other time we may introduce you the time of flight and LCMS instrument if there is a slot in this or in the next schedule of the FDP. So here is a quick look at the top. This is a very high-tech detector. And
ESI chamber through which separated compounds or sample come in this the loop through this, and here is the ESI chamber where ionization takes place, and further it pass to the quadrupole, hexapole, and their MSMS analysis can be done in collision cell. And further there is accelerator, so ionized molecule get accelerated to the top, and on the basis of thin kinetic energy is applied, and that is separate.
Thank you, Nitin. And now one more exciting instrument we are going to visit, and that is FESEM. So we are visiting FESEM. Field emission scanning electron microscope. Again, one important instrument which is being used very heavily, and Archana is with us. She is operating this instrument, so she quickly will cover the instrument. Will introduce instrument to you. What are the things we can do on this?
Hello everyone, this is Archana, and here you are looking. This is the field emission scanning electron microscopy machine. Here it comprises of the main part is gun. This is the field emission gun. Here the source whatever used for the this electron gun. Here we do a scanning technique. It is basically imaging technique where electron is used as a light source, like a source. So your sample is scanned throughout with the electron gun to get an image. And for that, we use a sample holder called stub. It is like that. And this stub is then put into the small chamber and then goes to the higher chamber. And the vacuum is created so as to get the electron field, and we get the imaging.
This is, there is a specialty about this. This is called auto loader. Okay, this is a very important facility because with the help of this, you can insert the sample or you can place the sample in the instrument, one can say remotely. Otherwise, you have to open this door and then you have to load the samples in from the front. And in that case, you will break the vacuum. It will take a lot of time to create the vacuum again. But all those things can be avoided. And with the help of this autoloader, we can insert the sample one by one. Yes, go ahead. And you can see, there are different detectors, which is one important is the EDS. Called from the Bruker, again X-Flash, and what result it gives, we will show you on the screen. Okay, next.
A few images taken from the microscope. This first is butterfly wings. This gives you different types of diffraction techniques in the wings.
So this is, very versatile instrument, I would say. Different type of samples we are receiving from the material science, from the biology, from the chemist, from the physicist, even from the dentist.
Yes.
Okay, and even we had some rope samples, the rope manufacturers, to see the texture of the rope. So n number of samples, n type of samples we do receive. As you can see, this is the butterfly wing. So someone is working on this butterfly wings, and the color, magnificent color of the butterflies are due to diffraction, reflection of the light, and that can, because of this structure, you can study the structure with the help of APS instrument.
These are whole nanoparticles. We also give, especially this machine is very much specified to go for nanometer scale. One of the best example is gold and tin. This is gold nanoparticles which can be measured up to 1 micron and even to 500 micron to 400 micron. The latter we have automobile samples also where they do a particular thickness. They deposit some layer and they need to check the thickness. So we take a cross section and we check the thickness of the layer deposited. Then we have other samples from biology itself. It is the mites. These are mites. They are usually found on the insect. These are very minute in size. So they can be also studied for their anatomy and other clarification or distinguish how they are classified into their classification, dorsal and ventral side. And also this is a leaf sample. We usually cannot see the leaf sample so clearly, but FESM gives us the clear look. This is also a leaf sample. Leishmania cell, bacterial cell. This is a onion root sample. Onion root sample. This is CNQ tubes. We can see clearly the deposition on the CNQ tubes here. This is a fat sample. These are the normal sample we get in our silkworm. You know, when the silk are deposited, you can see the deposition which gives a shiny material due to this material.
Gold particles. Okay. Now, as I said, we have a EDS detector. Okay. Now, Archana will show us the some EDS reports.
This is one of the EDS report of this of Zn particles. What EDS does, it gives us the elemental composition based upon this diffraction technique. Here the X-rays are used so that we can get a K-alpha value of the elements, and then we get a tentative distribution of the elements in your sample. So this is one of the parts which are taken from the sample, and it is only done with the live sample. We cannot take on the images. We need a live sample for the diffraction. We get this such type of diffraction graph from a diffraction pattern.
So you can see the elemental distribution at a particular point. That is very important. So at a particular point, you can see what kind of elements are there and what is the abundance, what is the percentage of that particular element on this. So
graphical and tabular notification.
Even tabular also you can get.
This gives you the tentative distribution in percentage of 100 of the elements in your compound. Also, we can also map the compound with different colors.
Yeah, so this is the mapping with the help of elemental analysis. So each element has got different color. So immediately you will come to know, say for example, green is say Cu, or some orange is some different say Mn or something like that. So this is how the big facility we do have. And all of you are welcome to use this facility. So Archana, thank you. Let us have a quick look at our office and how you have to submit the sample. So this is the instrumentation facility office, and this is the software we are using actually. So for example, now Atul will show us how to log in into the system. Okay, so for users, you have to go for the users, and then you put your mobile number, and you do have some password XYZ, whatever it is, and then everything is very transparent and very easy. Let us have a quick look at confocal microscope. Neha is there; she will show you some quick things about. Again, this is a confocal microscope. This is also very high-end instrument. Now Neha will show us some images through the confocal microscope. You can record the video also, for some interaction. Yes.
This is the Drosophila head. You can see the Drosophila head. We have done the imaging with the DAPI and bright field image. Then these are the glands.
You can see the DAPI nuclear stain here. This is a sample of tooth. Here live and dead cell imaging is done. These are the actin filaments. These are the bacterial film line. These are the images of neuronal cell line here. This is a few examples. And you can see the nuclear staining also with DAPI. Okay, this is the image. DAPI, this is FITC, this is red.
Okay, thank you, Neha. So I hope you have enjoyed the tour. Let me see whether I can switch the camera and show. Okay, so thank you very much. I can address some few questions if you have. Hello.
Hello. So, dear participant, have you any query or any doubt about this facilities?
Sir, if I have to give a sample for NMR analysis, like I have to do H1 NMR, so what are the pre-requirements? Do I have to make a selection of a solvent, or how I have to bring it in a dissolved form or a pure sample?
Okay, I think Snehal will address this question very nicely because she has to, face these issues every day. The question is, what are the prerequisites for the submission of NMR sample? So whether
sample should be soluble in. So your samples should be soluble in the solvent. Solvent chloroform is there, DMSO is there, D2O is there. So it should be soluble in solvent. Then it should be a diamagnetic in nature. And for liquid state, this condition is required. And the sample quantity, we have to add 0.5 ml solvent. So 5 milligram needs for the proton analysis, and for C13, 10 milligram is sufficient. Actually, it depends upon the molecular weight of the samples also. If it is a high molecular weight compound, then 10 milligram, up to 10 milligram, it's sufficient.
If the compound
solvent, another question was the solvent. Solvent, actually, you have to decide. CDCA3 is the normal solvent we are using. But otherwise, you have to choose and you have to check solvent solubility of your sample in your lab, which is very important. The sample should be soluble, 100% soluble, and then only you will get a good spectra, right? So that's all. Any other?
Suppose I find that it is soluble in a D2O. So I have to bring it in a dissolved form, a sample dissolved in a D2O.
No, actually we are using the deuterated solvent for the NMR analysis. You have to just check the solubility of the compound in your lab. You can use the HPLC grade solvent or GC grade solvent. But here we are using only a NMR deuterated in deuterated solvent.
So point is that you have to check the solubility at your end. You can submit the solid sample, and then we will prepare the sample as per your requirement. Okay, so as per your solvent requirement, we will prepare the sample, and then only we will insert it into the instrument. So you have to check the solubility, but sample will be prepared at our end. Few samples are very costly as you know. Okay, any other thing?
One more question, sir.
Sir, sir, apart from proton and carbon-13, is there any possibility of having 15 nitrogen NMR also, sir? Nitrogen 15 NMR?
No, can't hear you. Bit loud, please.
Yes, sir. is it possible to have nitrogen 15 NMR also?
Yes, yes. We do. It is tunable probe as I showed you in the presentation. So you can have, you can tune to phosphorus, nitrogen, silicon also, sometimes aluminum, fluorine, which are not very common, but yes, we can do that.
Yes, sir. Thank you so much.
Yeah.
So there is one more question is there from Nilima Devi. For electron microscopy, how do you send biological sample? How much quantity and in which form?
For APSM?
APSM.
Yeah, okay. For APSM, your sample should be non-magnetic. That is the first requirement. Then quantity is not the great deal. Actually, it's a microscopy. So small quantity of the sample is good enough. In fact, it should not be bigger. For example, sometimes dentists come with some, bigger samples or something like that. So I think 2 mm by 2 mm sample would be good enough. Less than 5 mm, right? So it should be 5 mm by 5 mm, less than 5 mm, and non-magnetic is the criteria. Anything else?
Okay. Thank you, sir. Any other question?
Yes. My question is in line with Nilma Devi's question, sir. If I'm interested in doing FDCM of my organism, and if I dye my sample, it may affect the architecture of my organism or a part of the organism. And if I don't have the CPD facility, what is an alternative to submit the sample?
Can you be a little bit loud and clear? Because we could not hear your question. We are through mobile actually. So there is a bit of limitation, but I believe your question is with the, again, can you repeat please?
Yes, sir. If I am interested in doing FDCM of my sample, and you accept the sample only in dried form, right, sir?
Yes, yes.
So if I don't have the CPD facility, so if I go for air drying of my sample, that may affect the architecture of my sample. So in what form
yes, definitely. I mean, when you will dry your sample, actually, it all depends on what parameters you are looking for. But definitely, it will affect on your sample. There are some bio attachments for FESM. Unfortunately, we don't have those bio attachments. But with the help of bio attachment, you can, scan the biological samples as the forms those are available in. I hope it has cleared your query, right?
Okay.
Anything else I can add up?
Yes.
Hello.
Hello. Any other question, please?
Sir?
Okay.
If we are going for publication, can we use the interpretation that you're going to give it to us?
No, we are very clear about that. I mean, we are the facility which will provide the scanning of your samples. So any interpretation, any special preparation, we do not undertake. Because our this much, I mean, we do have a lot of load. I mean, you can see 3 of them are working very hard, and this works 24/7. So our scope is limited to the whatever the sample you will submit, whatever the parameters you have asked. Like for LCMSMS, you have to give the what are the parameters you have to keep. Same is the case with GCMSMS. We will just scan the samples as per the parameters and requirements given by you. We won't give you any interpretation or any other alteration in your methods, and method development is not possible. Am I clear?
Yes, sir. Thank you, sir.
Yeah.
So, thank you.
Anything
sir.
else?
Anybody want to ask you?
Anybody else, or should we conclude?
Yes, sir.
Sir, it was a great tour, sir.
Yeah, thank you so much. I think मराठीमध्ये एक म्हण आहे, प्रत्यक्षाहून प्रतिमा उत्कट. म्हणजे तुम्ही इथे येऊन पण जे पाहू शकणार नाही कदाचित,
Yes, sir.
ते तुम्ही या टूर मध्ये पाहू शकलात असं मला वाटतंय.
Yes, and in a very short time, sir. Thank you so much.
Yes, yes.
And your entire team, yes, sir. Your entire team, we congratulate, sir, and thank them.
Thank you so much. Thank you for your presence.
So, it gives me great pleasure to thank our director of CIF and chairman of board of study, Dr. Kumar sir. Yesterday they told me that is we can arrange the tour and we can show the facilities. So it's great opportunity for us also. That is in our this faculty development program. You have participated and give your virtual tour also. Sir just mentioned that is those things we are not seeing directly if you visit that, but we can see in this visit. So it's very great thing. So on behalf of Ramakrishna Maharaj College and Faculty Development Program, all participants, I thank Dr. Kumar sir as well as Pradeep Kharade. They explain all this NMR about sampling and all in detail. And her colleague Sheetal, Snehal, they also explain very well. Also they show us LCMS with the help of Nitin and FPSCM with Archana. Archana is our student.
Okay. Good.
Research Center. So it's I'm very happy to thank you all for this your cooperation. Thank you very much, sir. So now the session is over. So tomorrow we can start our session at 9:30 a.m. We have 3 lectures tomorrow. Tomorrow is your last day, and in this last day, our first session starts at 9:30 a.m. exactly. And Dr. Milin Kulkarni from CIMET, they are going to give talk on nanomaterial, nanocomposites and their characterization. Second section is start at 11 a.m. and Dr. D.T. Dhawale, ex-head of Department of Chemistry, Central University, and they're going to give talk on characterization of NMR, characterization of sample by NMR. And the last patent after lunch break, there is a MCQ test from 2:30 to 3:30. 1 hour MCQ test is there. And after over of MCQ test, we start 3:30 our validity function. It's within half an hour we can complete that. So those who want to give their feedback in the validity function, please give their names. That's why we can arrange that here and we can make the schedule of that function. So thank you for all. Thank you. Have a good day.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
Thank you.